Good morning, everyone. Great to see so many of you have returned, so that's great.
Okay, good morning, everyone. First of all, I'm very happy to be here. I really enjoyed yesterday's session and all the very inspiring presentations. I also thank the organizers for selecting me to give this sort of keynote speak. It's a topic that you can talk for years, and that's the biggest challenge, actually, to fit myself in 20 minutes. So I ask kindly Martin to stand up, grab me, and put me in the chair if I exceed the limit. So I was wondering what we could say about this topic, and the first thing that came to my mind is my favorite Argentinian writer, José Luis Borges. Probably some of most of you know him, who didn't say that actually, but he liked this expression, that knowledge is memory and novelty is oblivion. And I think this is very insightful because at the very end, what we learn is not something that never existed, but it's something that practically happened and we realize somewhere in time and space that we came across to that. And I think that you will, what we heard about artificial intelligence, what we will do in the next years probably is not rediscovering the wheel, but revisiting very established principles. So, to me, the holistic risk assessment has the foundations in the One Health approach, because One Health approach combines human health, animal health, environment, and you may see all the challenges in the surrounding boxes, circles, like societal challenges, climate change, deforestation, biodiversity, things that we've been discussing for many years, disease vectors, livestock problems, security, food security, different consumption habits, and all these challenges are linked to keywords that we are using in these projects, because we talk about resilience, emerging risks, drivers, early warning, food supply chain analysis in order to understand where the risks, and particularly the emerging risks, come from. But also, I would like to add to our discussion today the combined exposure. So the combined exposure to multiple chemicals plus biological hazards. And at the very end, something that is very, very important, and I think that Jean-Marie will talk more about that in the afternoon session, the cost-benefit. Because we have to make decisions that reduce the risk, but none of these decisions is without any cost. One holistic risk assessment approach is practiced through the biohazard panel of EFSA that I happen to be member and vice chair for the moment. So when we receive dossiers about the decontamination agents and their efficacy in reducing bacterial contamination, we are actually having, as a standardized way, these terms of reference. So we not only evaluate the efficacy, which is the job of the biohazard panel, to test the log reductions caused by the decontamination agents compared to the water control, simply to washing a carcass, for instance, but we are also assessing, not ourselves, but the contamination panel, the toxicological safety of the substances, the emergence of reduced susceptibility to biocides, so selection of adapted clones or resistant clones due to the treatment with these substances, plus the risk related to the plant effluents in the environment. So it's a holistic approach that starts with the efficacy as the central player. But if something is efficacious, but at the same time has toxicological implications or causes burden to the environment, then theoretically the dossier is somehow negatively assessed. Now, regarding this balance of cost-benefit, and this combined exposure to chemical and biological hazard, I think we are exposed to well-known dilemmas so far. And I have picked up some examples. So nitrates in cooked meat products. I mean, if you discuss with many people, especially nutritionists, will say, get the nitrates away from our diet. But if you talk with a food safety expert, like we are now, everybody will say, but what will happen with the Clostridium botulinum? So we need the nitrates to control the spore formers in the cooked meats products. But at the same time, we are a bit concerned about nitrosamines formed by the contact of nitrates with the organic matter. So it's a cost safety, toxicological and microbiological safety balance. Chlorination in fresh produce. We had a very nice discussion during dinner yesterday that on the one hand, chlorination is mandatory to control a good microbiological quality in water used for washing operations for vegetables. But at the same time, there are indications that if you maintain a very high level of free chlorine, the contact with organic matter may lead to the accumulation of trihalogens, which is a toxic factor. But anyway, we cannot live without chlorination, at least for the moment, for the fresh produce safety. And finally, as I will present later on, thermal processing of tomato paste products. I mean, you have to sterilize and remove the microbial contamination, but maybe if you overdo it, if you over-process the products, you may have accumulation of furans or other products, toxic products that may be of burden for the health of people. There is a very nice paper by a PhD student of my long-time colleague that is not longer with us, Kostas Koutsoummanis and Martin Nauta. So it's Leonardo Stathas, probably you know him, a very good person, very promising scientist, who introduced the cost-benefit in sampling to prevent disease. I would like to stay on this graph, which shows clearly that the more samples you are analyzing, the higher the predicted reduction of the disease. But what is the limit? Who makes the decision on what cost criteria you will decide how many samples you will do in the official controls in order to achieve the expected reduction or the detection of the biological hazard. So we're talking now about risk reduction with the help of the sampling, and I would prefer to be honest, instead of talking to absolute risk values, to talk in relative risk values, because to me this makes more tangible the impact of interventions, that if you make one intervention over another one, then you have a 10-fold or a 100-fold reduction in risk. And then if you budgetize this option, then you can make a more informed decision as a manager. This is the courtesy of Yang Tai Liu, a young professor in the University of Shanghai. I was there last week, so I won't go into details, but this is how they see it in China. So you have the food supply chain, and you see the plant, the animals, the environments, and the human. So they consider all these pathogen, chemical contaminants, and their interaction with plant, animals, and human body as determinants for the final risk, either starting from the end, up bottom, or starting from the beginning as a bottom-up approach. And everybody needs to find their role in this continuum of the food supply chain. So we are living in a complex ecosystem, but it always used to be a complex ecosystem. That's why I said that knowledge is memory, actually. Now we have the tools to revisit the same things and probably get more deeper insights. Now, as a microbiologist, I cannot resist starting with some microbiological challenges. And I think the most important challenge is the variability, which exists everywhere. It makes our life beautiful because we are equal, but we are not the same. And the fact that we are different, it helps us mating, have nice discussions, have nice dinners, have nice parties, because we look for the diversity. But at the same time causes some headaches. I would like to drive you back to beginning of 2000. I happened to be in the Institute of Food Research in Norwich, and I participated in an experiment with the so-called technique of gel cassettes. So they put microcolonies under a kind of confocal laser microscope, and they monitor the 3D structure of immobilized colonies in an agar. But the most revolutionary, that was 26 years ago, is that they had tagged with a GFP the gene of, this is Salmonella microcolony, that is associated with the acid resistance. The decarboxylase of lysine gene that is called CAD-B, that produces cadaverine, for those that are microbiologists. And eventually, they observed that this gene is expressed in the center of colony, because in the center, you may have a concentration of cells, accumulation of metabolic byproducts. Most of them are fermentative byproducts. And this induces the adaptation to acid stress. So that's why the risk of the acid-resistant cells is in the middle. But this colony is in the food, and when you eat the food, you eat all the cells. Maybe the surrounding cells might not be of high risk for you, but the central cells might be quite resistant to the gastric acidity. And when you do the analysis in the lab, but you don't go into the molecular stuff, you are counting the total number of cells, but you don't know which of these cells are of higher risk. The second thing, I happen to have a very competent PhD student, Mariana Arvaniti, who deal with the viable but not culturable cells. That's a great challenge for underestimated risk, because actually we think that these cells are dead because we cannot culture, but unfortunately they are alive and they can regrow if they find proper conditions. And these cells are variable. This is a typical example. So in a population that is exposed to peroxyacetic acid, because we did a lot of work with peroxyacetic acid, then you have cell division, but you see that the number of generations here are symbolized by different colors, and not all cells have the same color, which means that not all cells are in the same generation, individual generation time. This is, of course, a work that was first initiated again by Kostas Koutsoummanis, and I'm really happy to be very close to him and inspired from his work. So these are cells after exposure to peroxyacetic acid, and when we, apart from monitoring them and capturing their variability, we also convert these pictures into numbers. And this is something very important. So these are the generation times of the different generations, consecutive generations. These are the control cells, and I will guide you directly to these cells. So these are cells that never exposed to peroxyacetic acid. And this is a normal picture. As we proceed with generation times, with generations, the generation time squeezes because the initial memory of the cell for the previous environment actually diminishes. And this is the normal pattern. But what we came across was this trend here. So we have a bimodal distribution. So we have cells that are having practically very low lag time. They are fastest recovering fraction, short lag, which means high risk. But at the same time, we have cells that have been severely injured, and no matter how many generations we are after, they maintain a very extended lag time. So what would be our risk assessment? The risk would be higher from these cells because this actually will overcome the previous exposure to peroxyacetic acid. But we need to know, because 30 ppm is a really high, I would say a bit sublethal concentration that may happen accidentally in a processing plant. And eventually it may select cells of different physiological state. So one of the challenges is to be able to quantify the impact of the environmental and industrial processes on the behavior of individual cells and the variability, especially after exposure to stresses, because this is where the risk is diversified. That is a very recent experiment we also did in the lab. So I kindly ask our colleague to initiate the video. Exposure of listeria to bacteriophages from a Dutch company commercializing the PhageGuard solution. So as you may see, within a 10 hours period, we have a GFP transformed strain of listeria, and by the invisible, apparently, contact with phages, which are 100-fold times higher population than listeria in this field, they come in contact with the cells and the cells start to die off. But not all cells die off immediately, because then we should have, if you look at this picture here, we should have a drop of the population down to zero. And some of the cells are not affected. So now, this is a complex phenomenon. Right? How many phage particles attack the cells, or how many they are needed to attack the cells in order to burst the listeria cells? How many of the listeria cells may be able to resist because of their cellular morphology, their cell wall characteristics, and eventually what is the dynamics of phage particle released from lysed cells, and depending on the mobility of listeria, here are immobilized cells, in a food product it could happen, they may come in contact with new listeria cells. So we have a lot to study by making a simple experiment about the dynamics of inactivation, for example, of listeria, even by a lytic agent. Now, I won't go into details, but I want to catch here the following thing. We have strain diversity and population heterogeneity. Strain diversity is what was described yesterday by Lauren Alteo very nicely. So we have transposon, plasmids, prophages, elements that diversify the strains, create SNPs, they make different strains behave differently. But we have also heterogeneity within the cells of the population. This is what I was showing before. So isogenic cells, but with the genetic and phenotypic noise. So we have cells behaving differently, not because they are different strains, but because they are different cells. If you take this picture, you can fit it here. Why? Because This is one source of variability, genetically explained, but we have other sources of variability. Product characteristics. Not all products have the same pH, even if they are standardized as batch from the industry. Not all products are exposed to the same temperatures. Not all products are exposed to exactly the same processing characteristics. Although we do that, we aim for that, but this is practically non-feasible. So, how is this translate? That's why we need a probabilistic way of thinking. Of course, many of us here, we are familiar with probabilistic way of thinking, but I can tell you not everybody. So we have to express ourselves in terms of probabilities, because the risk is a probability by definition. So we cannot talk in absolute terms. Huh? And this is a typical example how probability, for those that are not familiar with Monte Carlo simulation, this is how the probabilistic way of thinking may help us improve our risk assessment. So this line that moves like a snake, okay, is the behavior, the dynamics of the microbial population, because variability in product characteristics, time, temperature, bacterial physiology, like the videos I showed you before about single-cell variability, plus the cooking preference and the serving size. I don't know if the social scientist colleague is with us today, but this is very important. The cooking preferences, log reductions caused by the consumer, serving size consumed, okay, define the amount of the amount of dose that will be ingested by the consumer, and depending on the dose response models, eventually determines the risk per serving. But I would say that here there is a whole discipline behind that. DALIS, CALIS, relative risk reduction. So there is a lot of work to be done in order to convert what happens, what we eventually describe with our wet science tool, with our bench tools in the laboratory, into the risk assessment. And this is practically what happens. Not all packages of one batch, like 100 packages, will go at the same temperature. So the packages are exposed to different temperatures. This inevitably happens when we go to the supermarket. This means that they can be represented by a histogram, and instead of talking about discrete variables, we can represent the histogram by a continuous probability distribution. And then you may replace the example of temperature by time on the shelf, product characteristics, etc., in order to reproduce the line I showed you before that varies up and down, describing the exposure to the consumers. And why is this important? Because this would be our prediction based on average estimate. But if we have variability, for example, in pH and temperature, then the actual prediction will range between these two lines. And why is this important? Because if we have a microbiological limit of 2 logs, for example, for Listeria, then in this case, the actual time needed to exceed this limit varies from this place to this place. And there is a portion of products that will exceed the limit much earlier than what we expect, relying on the average. And as Marcel Zwittering used to say, devil is at the tail. Huh? So who is going to eat this portion of products? And of course, another important consideration is uncertainty. We heard a lot about that. This is what happens in Monte Carlo simulations, sampling from different distributions. But although sampling can happen many times, the question is, are we sure about the shape of the distribution? Another thing important for microbiological, but I would say also for chemical risk assessment, are we sure about the dose response models, especially for the chemical risk assessment? Not that the microbiological is the easy one, but I think for the chemical risk assessment, especially the case of combined exposure, selecting or reflecting, mathematically speaking, the dose response model is very, very important. So our uncertainty about whether we are using the right probability distributions and whether we are selecting the proper dose response models is a challenge. And finally, big data equals to big challenges. I think that was a clear message in the consortium yesterday, especially in the yesterday presentations, that we have a lot of data. We have generated a lot of data, and we will still generate data. But to me, the challenge is, like EFSA says, from data collection to data connection. The most important is data sharing and connecting data. That's very important. And I want to show you quickly this example of a dashboard that we have created and was shown in Spets's meeting in FoodSafeR. So we decided, as part of a Greek project, to collect data for different biological and chemical hazards. The biological hazard was Salmonella listeria and some spoilage organisms, not as a hazard, but as an information about hygiene. And the chemical hazard was polycyclic aromatic hydrocarbons, nitrosamines and nitrates, and we want to expand that to pesticides. So we tried to collect samples from different industries, be able to show them relative to the picture of the different companies. So we wanted to ask the companies to see themselves in relation to the other samples from other companies that their information was disclosed. These are multispectral reflectances, which could be somehow also stack bars of relative abundances. So if we have, for example, the relative abundance of different groups of the microbial consortium and be able to have a complete picture of microbiological, chemical hazards, and microbiome. This is the map that shows the spatial distribution. But the most important, as you will see in a few seconds, is that you can filter data out of this dashboard, and this is something that was also shown yesterday from the CEFS database and other applications. And based on the filtered data, you can perform some multivariate analysis, like principal component analysis, like partial least square regression, or supporting vector machines, whatever algorithm you can call from the background. And you can see the example here of principal component analysis. I thank the colleague for running the video. That helps a lot because I have to finish in 1 minute. So this is the correlation of total viable counts predicted by multispectral data. And this is another important asset, that based on the product characteristics stored in the database, we give direct predictions of the growth of microbial hazard. Here is the example of listeria. So it's fully interactive. That's why I mean that it has to be interoperable, shareable, so that people can enrich this database and at the same time perform mathematical activities or statistical activities like machine learning with the data filtered out of the database. So if you select, for example, specific characteristics of products in the database, you see the lines here are updated. And if you click this button here, if we can run the video a little bit, then it opens another growth prediction tool that we have developed that is called Growth Predictor. So we can please proceed to the next slide. Okay, and the example that Lauren presented yesterday, in FoodSafeR we had the opportunity to collect many different listeria strains, and now look at this point here. We had the opportunity to collect many different listeria strains. Some of them were characterized as persistent because of frequent repeated isolation over a period of 2 years, and perform some phenotypic assays, MIC of different disinfectants, minimum inhibitory concentration apparently, biofilm formation capacity, but at the same time through whole genome sequencing, be able to identify a list of potential biomarkers associated with persistence. So we annotated these strains, as you see here in this column B, as persistent or non-persistent, and we were able to correlate, was our ambition at least, to correlate the genotypic and phenotypic data with the probability of this strain behaving as persistent or not. So this is a challenge that combines so heterogeneous variables in order to explain a specific phenotypic characteristic. So we developed another machine learning software tool that offers the option to apply different machine learning algorithms, do a pre-processing of data, so there is a lot of parameterization, but this can be done directly on site if you can click on this link. And eventually, we ended up with these confusion matrices where you see that we try to correlate, based on genotypic and phenotypic data, the persistent characteristics of listeria that we used in order to train these machine learning algorithms. Apparently, the problem with these algorithms is the data. The more data we accommodate, the better for us. Yes, I'm finalizing, sorry. So, AI. We talked about a lot of AI. I think we heard yesterday the opportunity of AI to be able to correlate pictures of plates with bacterial names. So this is the YOLO-based application from a group from Purdue University in the States, where they tried
to somehow annotate the debris in a food sample and the microcolonies in the microscope with the AI and object recognition algorithm with deep learning in order to be able, by watching directly sample under the microscope, to understand the type of microcolonies developed, present. So this is the industry of the future. I think this is the big challenge in the 21st century. Why are we doing all this? Because we want to harness the data that we have available, be able to monitor the food supply chain, and using the machine learning algorithms, but keep a note on that, to be able rapidly and quickly assess the safety or the quality status of the foods. So practically speaking, it's like taking a spectral image, having a whole genome sequencing data set, having a phenotypic list of properties, be able to convert that into useful decision support information. To me, this is the ultimate challenge. And as I said before, this is a work that we do in collaboration with Michel to correlate the presence of furans based on multispectral data. So we capture multispectral images of tomato paste, and through machine learning algorithm, we are trying to estimate, and this is our validation results, very promising, I must say, about the levels of furans in different products and what the machine learning algorithm predicts. So to conclude, we started from information. We went to data. Data offered us knowledge, but now the challenge is to convert this knowledge into wisdom. I think this is the ultimate challenge. And the most important is not to forget this, because I think this is the key message. This is my favorite slide, that our microbiological background and our predictive modeling background relies on causality. Certain stimuli cause certain results. AI is the smart and good-looking guy that gets all our girls away from us, or all our men away from us, depending. So it's efficient. It's very rapid, it's very efficient, but it's away from causality. So it's a black box that gives you very efficient, very important answers, but doesn't explain you why. So we have to find a balance between causality and efficiency. Thanks for your tolerance.
Maybe time for one question. Let's make it a question and a comment that goes for everyone.
Thank you very much for this overview. It was very good. Lot learn from it. And you were, I'm not sure. Is it? Yeah. I was wondering, when you bring in a diversity of the development of these risks, is that also a consequence for having food safety standards? Because then it can develop in human bodies in various ways, whether the condition of the body, etc. So you were referring to social sciences, but there might be a big issue still to be developed.
This is a very important question. If I understood the concept behind that, I think we have to always rely on the worst case scenario. I mean, the legislation relies on very safety margins, and there is a kind of rule that if you know the, as we call the no adverse effect level, which is the minimum level that starts causing a problem in human health, then the legislation, the regulatory limits should be at least some magnitudes lower than that, so that it protects the consumer. The question here, and this is something that receives a lot of discussion, is how much lower should be this limit in order not to throw away many foods that are not harmful for humans, but at the same time respect these individual vulnerabilities of different consumers. So I think this is where science needs to jump into, and this is where actually we need the convergence, the interaction between social science, regulatory science, and of course the classical toxicology.
Yeah, thanks. Clear.
Thank you. Moving on.
Nathan. I have the pleasure to announce the next speaker, which is me. and I hope you have time enough to listen to my long CV, which I've prepared. No, don't worry about it. My name is Rudi Kriska. I think some of you know me, and I'm co-coordinating this activity, FoodSafeR, with Martin Wagner. And now I would like to switch gears and jump right into the depth of the subject on advanced analytical methods for chemical foodborne hazards. Certainly this talk is pretty much structured around the FoodSafeR project, as you can imagine. I'm also very thankful to my co-authors and to the entire FoodSafeR consortium, who have all contributed to my presentation today. Is this the next slide? Is this supposed to be like the green one? Okay. All right. Okay. Martin Wagner has already pointed out that within the FoodSafeR project, we also have an international advisory board. I'm actually very thankful and proud to announce that also James Lindsay, former USDA, is here, also online. Our chair of the International Advisory Board, John Leslie from Kansas State, he's on the line. These members, also in conjunction with some members of the Food Safety for You consortium, for instance, Veronica is here. We have also great collaboration with them. We have actually summarized their input with respect to pretty faces, you have seen this before. We've summarized this in this paper, also already announced by Martin in his presentation, with a pretty impressive number of views. This FoodSafeR perspective on emerging food safety hazards and associated risks also summarizes the risk agent. Like we have a table, food safety hazards, as discussed in a FoodSafeR workshop, and particularly in Barcelona the other year at IRIS premises. These are those risk agents which were particularly pointed out by our advisory board members, including plant toxins, algal toxins, mycotoxins, certainly toxic elements such as arsenic, cadmium, and then a range of chemicals, including processing contaminants, including forever chemicals like PFAS, pesticide residues, etc. How can you cover all that in one European Union project? There's no way of doing that other than presenting some of the information available on a platform. As I've mentioned before, there's a very useful platform also for food safety for you. We have more focused on the emerging issues, and what we have actually done is to identify the drivers and stressors of these emerging food safety hazards and associated risks. And you can also see this categorization into chemical, microbiological, physical risks. And to some of those which actually with a red asterisk, we have particularly carried out dedicated case studies. That is particularly true for process contaminants, illegal addition of toxic chemicals, also chemical of biological sources, and also antimicrobial resistance. That information can all be found on our FoodSafeR platform. If you are not a member yet, please register right now, and certainly all the data which we have created or have been creating will also be available to the European Food Safety Authority. So our project is pretty much structured around chemical hazards and risk on the one hand, going to be presented now, and the tools for analyzing and identifying those. The microbiological part will be presented in the next talk by Martin Wagner. So first one on combating illegal additions of toxic chemicals. So there's a lot of massive food fraud going on in the area also of spices and food fraud in this particular arena. Advanced mycotoxin monitoring, where we actually have selected those areas in Europe where there is particular grain harvest, also to improve prediction tools. We have also immersed ourselves into process contaminants such as furans, and certainly also on mycotoxins, and particularly also the fate of these toxins during thermal processing, utilizing novel technology, which I'm going to present in a minute, and also advancing bioanalytical sensing techniques and diagnostic tools, particularly for pyridine alkaloids on the one hand and tropane alkaloids on the other hand. So you can see a lot of things going on, so please fasten seatbelts for my speedy presentation and listen to my broken English, which I will present in the next minutes. This is one way looking at it. And the other way of looking at it is actually looking into drivers, right? So that was nicely done by Lisbeth Jackson from UKent, who has also been serving as a work package leader, who has actually, based on the participatory workshop within stakeholders, International Advisory Board input, identified these drivers of emerging hazards and associated risks, social drivers, technological drivers, economic drivers, environmental contaminations, so-called environmental drivers, and also political drivers. So actually, in total, 39 sub-drivers were identified. These are actually the major drivers. And when we categorize or kind of, when I guide you through the FoodSafeR project from the chemical arena, we can also look at the whole story from this driver's point of view. So let's now kick off this part of my presentation with the economic driver, food fraud authenticity. So that is clearly a topic which was also already addressed yesterday by Paul William. So you see illegal addition of toxic chemicals, lead chromate in turmeric, which is a very popular spice also in India. But there's actually all these emerging markets, and there are all sorts of sources from which this turmeric is originating from. So what Paul did was actually to look at this turmeric spice, which has numerous claimed health benefits, but lead chromate and fentanyl is actually used to give a more vibrant color. He has been dealing with portable spectroscopic devices, particularly with X-ray fluorescence devices, also with NIR and FDIR. And to make a long story short, because he has already presented yesterday anyways, he pointed out that machine learning is a tool which can be used particularly not only for the laboratory test benchtop XRF, but particularly also improving the field testing with a handheld XRF, particularly to determine the lead content as an indicator for the chromate. We also had several meetings on that at Queen's University, Belfast. So when we switch through to the next driver, legislation, policies, governance, you see here the EU Rapid Alert System for Food and Feed, RAFS, actually with the data of the last few years. You see pesticide residues, pathogenic microorganisms, mycotoxins, these are on the podium. Although the perception is that pesticides are more toxic than mycotoxins, which they are not. Pesticides are much less toxic, also not carcinogenic as mycotoxins, but that is a different story. Mycotoxins is the third most notified hazard, and that is also why it's justified looking into that particular view of climate change, into this particular range of contaminants. And when we look into into the next driver, environmental drivers, the mycotoxins, the natural toxin arena is particularly the one to be addressed. The worldwide contamination of mycotoxins was estimated by the FAO, the Food and Agricultural Organization, decades ago to be 25%. So we have challenged these figures. There's no original reference which actually clearly defines or provides information on how this 25% figure has been derived. As a matter of fact, that's why we came up in collaboration with EFSA and also utilizing biomine data from feed and also from food from EFSA, that we have indeed not the 25%, but up to 80% of the food crops are contaminated with mycotoxins at detectable levels, and that is clearly also thanks not only to the dramatic issue of climate change, but also because of advanced analytical methodology. The methods which have been in use to derive that figure were mainly based on tandem mass spectrometry, HPLC, coupled to UV, etc. ELISA immunoassays certainly on the rise, and that is particularly true for the feed arena. This is more the food arena. This is the feed arena for the biomine surveys. So this just gives you a flavor of the range of the analytical techniques. If you would like to advance this a little bit further, we have made an attempt also to look into infrared spectroscopy and also Raman spectroscopy as a novel tool to estimate mycotoxins, although they cannot directly detect mycotoxins because it's not sensitive enough. But through correlation of the matrix changes as a result of the infection with the fungus, you can actually quite well correlate these spectroscopic changes with the mycotoxin content. And these are some highly cited research papers. If you're interested, please have a look into those. The backbone of our own research has always been here at BOKU, multi-class analysis by utilizing the power of tandem mass spectrometry with this electromagnetic filter of the quadrupoles, where we have kind of optimized and brought it to the next level, a simple dilute and shoot approach, extraction, dilution, and analysis, which is actually, by the way, much more superior than any catcher's approach. So if anybody tells you that he needs catchers, if you have a good instrument, that's actually not required. We have realized 1,200 contaminants, including 500 mycotoxins and derived compounds in 45 minutes runtime. This was further elaborated by my PhD student Lydia Kenyarich and Michael Sulyok. This actually, she's also a great opera singer. She sang Edelweiss in Salzburg last year at the World Mycotoxin Forum. She's a great scientist and singer. It's a good combination. And she has actually brought these methods to the next level, being able to quantify, not detection, quantify at a reasonable level of small uncertainty, 931 secondary metabolites, including all mycotoxins in 11 minutes, with actually a very low limit of quantification, even for aflatoxins. That was also recently published. And then you see a plethora of substances, not only the regulated mycotoxins. You see moniliformin, you see coumarin, you see N-etins, which are cyclic hexadepsipeptides, which are also quite rapidly and often occurring as emerging contaminants, and actually some impressive high levels of some mycotoxins in some 4,300 samples, which were analyzed by utilizing this approach. It was already pointed out yesterday that the European Food Safety Authority has categorized emerging risks as either a newly identified hazard with a significant exposure, which could be a masked mycotoxins, where actually the toxin itself is bound to a glucose in order to enable the plant to detoxify the mycotoxin. This would be like then the combination of an advanced method being able to detect the masked mycotoxins and come up with these new findings, or a known hazard with significant exposure, and that is clearly an indication that a so-called emerging toxin does not only cover those toxins which are non-regulated. Aflatoxin is a very well-known regulated toxin, also the most carcinogenic compound we know of, produced by nature. And this new significant exposure as a result of climate change is another indication. Well, we are dealing here with an emerging hazard and a lot of associated risks. Climate change, plant breeding also here has an influence. And also a known hazard with actually different increased or decreased host susceptibility and significant exposure, such as with formonicins, is another emerging risk. We have identified back in 2005 the first scientific evidence. Franz Berthiller in my team, who provided the first scientific evidence for the natural occurrence of these masked deoxynivalenol coupled to a glucose, which can then be reconverted to the parent toxin after enzymatic conversion in the gastrointestinal tract, to be more precise, in the large intestine. And well, that was published in 2005. It took not even 20 years until the European Food Safety Authority came to the conclusion that besides acetylated also masked deoxynivalenol glucoside has to be included in the group tolerable daily intake. So some things take us quite a long time. So this is actually the glucosol transferase, which is important to facilitate this coupling to the mycotoxin to make it non-toxic, but as I said, in large intestine can be reconverted and then elicit toxic effects again. And coumarin is a co-occurring mycotoxin to deoxynivalenol, also produced by Fusarium. And it was just recently published that coumarin inhibits the detoxification of the mycotoxin deoxynivalenol through inhibiting this glucosal transferase. But it also inhibits the glucuronidase, which means the detoxification process going on in your body, in our bodies, is also inhibited. So that clearly indicates coumarin suppresses these detoxification processes in vivo in the plant and also in humans and animals. So co-occurrence of deoxynivalenol and coumarin enhances, so to speak, the toxicity of deoxynivalenol, if you may say so. And there's another interesting finding that deoxynivalenol is not a genotoxic, but colibactin produced by E. coli is, and deoxynivalenol actually exacerbates these effects. So, which means the deoxynivalenol itself is not genotoxic, but it makes colibactin obviously even more genotoxic, and that is why possibly we could think of enlarging or extending the definition of an emerging risk or an emerging hazard as by a known hazard plus new evidence for combined or novel toxicity. So this is just a suggestion here. Well, certainly there is also stressors like climate change. We have been looking into durum wheat, common wheat, tomato samples from different parts of Europe over the years to come up with new occurrence data, which we will provide EFSA, but also to get a better basis for big data long-term monitoring for new prediction tools, and also to provide evidence for the impact of climate change and actually to more accurate forecasting. Actually, a quite nice overlap with a project which I coordinated over a period of 4 years, some 7 years ago. My toolbox, where we also developed such prediction tools where you get then for a certain area a yellow or red alert for the formation of mycotoxins. The more occurrence data we have, the better we can refine those. This is a sister project of FoodSafeR, where we just recently provided the first evidence for the presence of aflatoxins on the field in Austria, not only in Lower Austria, by the way, in Austria. So, which means climate change has arrived. Aspergillus flavus can really also infect our plants. Aflatoxins is a risk now also in countries of moderate climatic region. And that brings me actually to this avalanche, to tsunami of media reports as a result of our findings. This was just 3 weeks ago. And that told me, and this was the message which I also got yesterday from Finn's great presentation, that it's not only about identifying and managing emerging risks, it's also about how to communicate the findings. That's another challenge, and I think we did quite a good job, and actually was a quite balanced information. Nonetheless, that remains communication remains to be a great challenge. How to identify emerging hazards against the backdrop of climate change? That is particularly done by high-resolution mass spectrometry. You have actually all these ions. Orbitrap can accommodate some 2,000 ions theoretically, which travel actually different radio frequencies around the electrode and can be separated quite accurately. And then you do a screening, for instance, by utilizing the power of stable isotope labeling assisted metabolomics. And by doing so, then you detect a plethora of other substances. It's not only B and A trichothecenes, it's also their acylated forms, it's also their glucosides, their triglycosides, their sulfates, and also their acidates. So there's a plethora of substances which you will only recognize, and which is also very important to look into all these substances to get a full spectrum and a much better and improved risk assessment by looking into those. Well, we also looked into process contaminants, furane in foods. There's not so much to be said about this one because there will be a presentation by Michele Suman and Alexander Schamann later on. Furane, as you know, is widely present in foods. Different heat-induced pathways induce furanes, which are possibly carcinogenic according to IRSC. You see also that in pilot plant industrial scale, furane was found, and the message was that in FoodSafeR, thanks to the great collaboration with Barilla, we have been able to study actually the fate of these furanes at a semi-industrial pilot scale directly at the premises of a big industrial player with headspace, solid phase microextraction, GCMS, gas chromatography coupled to tandem mass spectrometry as the state of the art for the determination of these furanes. Finally, I would like to present the plant-based products. So there are propane alkaloids on the one hand and pyridine alkaloids on the other hand. We've been looking into for the propane alkaloids which cross-contaminate these thorn apples, cross-contaminating cereals, the cereal harvesting on the one hand, and pyridine alkaloid in herbal teas, particularly in rooibos, a work which was done at Wageningen University, and that is was a really rewarding topic. I think we have been looking into these tropane alkaloids. There are about 200 tropane alkaloids, which will also be presented. That's the beauty of my presentation today, because it's will present the details anyways, but just to make a long story short, just in brief, daturine, soybean and maize, cross-contaminating, containing these tropane alkaloids, but particularly atropine. It's actually there is an EU limit also for grain-based baby food. There has also been recall of these tortilla chips, and that is something where clearly a method was had to be developed since there was no quick and simple method available before FoodSafeR. And it's and under supervision of Gerd Sallentin, they've developed actually a sample preparation using paper immobilized phase microextraction. It was also pre-validated for buckwheat and cereal samples at very low levels for atropine, and you will hear about that later. For the tropane alkaloids, there was also a validation which was carried out on a very nice Biosense work table. I really love that picture. So it was really a great job which WFSR has carried out in that. So congrats, it's. So pyridine alkaloids in rooibos. Well, I love rooibos every morning. Maybe I don't like it so much after your work, it's, but now we can actually detect those much better. There is about 4 of the pyridine alkaloids which account for about 70% of the pyridine alkaloids being present in rooibos. As you might know, there are these substances, there are about 350 pyridine alkaloids which are produced by Jacobeo Senecea weeds, which all share actually the functionality of two 5-membered ring system, which share a nitrogen bridgehead, which can also be oxidized, so which makes in total 700 substances. But for rooibos, there are actually those 4 substances, including these N oxides, which are of importance. And then you add another 30%, like the uncertainty that you end up with an estimate of what the level of pyridinic alkaloids in the rooibos might be. That was done actually here, particularly for rooibos. There are 35 pyridinic alkaloids which are commonly present, and by utilizing these as markers, these 4 compounds, by utilizing a lateral flow test design, they came up with a nice, alkaloid test kit, which was also validated after marker selection, then reduction of the N oxides, so that actually the antibody is also cross-reacting quite nicely for the N oxides, and then doing the immunodetection. Finally, a very nice citizen science exercise, I would say, which it has carried out here in collaboration with all these partners. So you see a very enthusiastic organizer, and that was done here, for instance, also at the premises of FOXI, of the Austrian Competency Center for Feed and Food Safety. This is my secretary, this is the accountant of Martin Wagner, and this is another accountant of Martin Wagner. So they have never been in a lab, other than visiting maybe a lab, and they did the validation, and that was actually a very successful exercise. It's another very nice outcome of FoodSafeR. Finally, social drivers, consumer behaviors, gluten-free products. So gluten is the most important poison, as you know, nowadays, although there I think about 2% of the adults having a gluten intolerance. It's a big story, but certainly there's a lot of suffering certainly as well. It's still a significant number. Well, then you have a shift from wheat-based product to maize-based product, which means that rather that you can expect deoxynivalenol in your spaghetti, in your bread, whatever, in maize-based products you can rather expect zearalenone, aflatoxins, fomonicins, or when you switch to barley you will expect the T2 toxin, HT2 toxin, which will also be presented later, but this is also done with high-resolution mass spectrometry, actually by utilizing the power of isotope labeling tracer fate studies. So what you do is actually not for DON in this case, but for instance, in our case, for aflatoxins, you spike your matrix, which could be your pasta, whatever, with the naturally occurring C12 backbone aflatoxin, B1, and you buy from the market a C13 labeled aflatoxin, which has C13 isotopes as a backbone. And then actually what you're having is a mirror image of the isotope pattern, and with our software, which I've presented, which actually is looking then for the degradation, what the beauty of this system is that any mycotoxin which will be degraded, so let's say the aflatoxin is degraded, also the C13 labeled compound will be degraded. And then you have the software, and this software will be looking into pairs of the C12 and the C13 isotope logs, and only if those both pairs occur, then you can be sure that the degradation took place. Why is it so complicated? Because in high-resolution mass spectrometry, about 95% of the data you produce is actually just garbage, electronic noise. So that's why you are utilizing this approach. Aflatoxins, fomonicins, and T2H2 toxins were studied and were not degraded during commercial pasta making. If you're interested in the outcome, listen to the presentation of Michele Suman later on. I'm not going to reveal that because I'm about to conclude my presentation. For each of the 5 major drivers of emerging food safety hazards, analytical chemistry plays a crucial role to manage those challenges. I hope I was able to demonstrate to you that tandem mass spectrometry for the ultra-fast quantification of multiple classes, because it's not only about mycotoxins, provides selectivity, sensitivity, and also the speed even for complex food and feed matrices. Stable isotope labeling assisted high-resolution mass spectrometry, the ideal tool actually to study novel conjugates such as masked mycotoxins, but also to study the fate of mycotoxins and other compounds during thermal processing. Indirect competitive lateral flow immunoassays for atropine has been developed within FoodSafeR. A novel approach for determination of ferritin alkaloids in rooibos based on a 3D printed biosensing toolkit was also introduced. And finally, portable spectroscopy, for instance, based on XRF combined with machine learning, was also introduced as a new tool, particularly for the handheld device for illegal addition of lead chromate in turmeric. That being said, this information is also again on the platform. For instance, under lifelong learning material, you find predictive modeling, you find the perspective of food safety hazard, you also find the information on the paper immobilized liquid phase microextraction and all the technique and technology behind that. This is to thank you all for listening, and I would also like to thank Martin for an incredible journey, these 3.5 years together. If you are not fed up with my presentation today, there is a chance that we meet each other again at the World Mycotoxin Forum next year at the Rotterdam Harbor. There will also be a sampling workshop there at the Rotterdam Harbor. At the moment you can actually register and apply also for presentation, and you get this early bird registration. So hope to see you there as well. Thanks for listening, and as I'm the chair, I kept within my time somehow.
20 minutes.
Thank you. Yeah. So there's no time for a question, but I will be around for coffee break.
Yeah.
And
otherwise, we have a 5-minute coffee break, and that's.
Yeah. And we will move on to the next speaker.
Yeah.
Martin, we are very much looking forward to your presentation. Same project, same topic, but dealing with the microbiological hazards. Please, floor is yours.
Thanks, Rudy. So I am pleased now to share some insights in our research that's not merely done in FoodSafeR, because we haven't had so many activities on method development in FoodSafeR in the microfield. But I would like to share some experience that we have gathered in the previous years. As you know, the development of microbiological methods had been speeded up in the recent years dramatically. There's a lot of pathogens that came into our spotlight in the recent decades. In 78, there was the first Campy enrichment broth developed. Listeria came to our mind as a foodborne pathogen in 83, because with the first outbreaks in Canada, the coleslaw, and then a lot of other pathogens. And by all these pathogens, there were developmental steps triggered to overcome culture-based, enrichment-based media. They were completed by selective plates, chromogenic media. Then immunological rapid technology was developed, and that was completed then finally with PCR, RNA, and DNA-based technology. And I think this story is ongoing. Nowadays, I show you some data on mass spectroscopy that has been replaced the identification of pathogens, I think, in most of the labs. This is what is the ideal world. If we work on a pure culture, we see these very nice colonies with a very clear phenotype, a very nice color, a halo, for example, here for Listeria monocytogenes. And this is what we see in reality. We see multifaceted phenotypes. We see commensals growing on the plate because a food environment is a very rich environment in terms of bacteria. We only have the pathogen. Here is the Listeria. This is the only colony in that plate that can be addressed as Listeria. So we're fighting, and this is, I think, a big difference to the chemical analysis. We're fighting in a very dynamic field to find the few pathogenic cells that are there. And we were always asking ourselves whether single cells do grow to populations, because the hypothesis is that from a single cell, we see a development into a colony. And that is not really the truth. We did this study already a couple of years ago, where we developed a device where we could transfer single cells, bacterial cells, Salmonella and Listeria monocytogenes, from a broth into a fresh broth, and then we incubated it, and we could see that depending on the phase from which these bacteria were harvested, lag, log, or stationary phase, only a certain percentage of these cells in that state were really viable. And this is what gets over to Panos, what he's saying. We have a lot of physiological states in between what is a living cell and what is a dead cell. And this has become a very nice topic of research in the meanwhile. Growth is stress, and we shouldn't forget about this. This is what's an observation that we did a couple of years ago. Even if you have, this is PFG, at that time we couldn't sequence the whole isolates. Even if we have no changes, obviously, in the genome through these passages, we see that the phenotype changes. Here, this color stain gets lost over these passages, and in the meanwhile, we are capable of analyzing sequence in these isolates and looking into what is the reason behind it. And this was a cross project from FoodSafeR, where we could show and publish that especially Listeria monocytogenes, the color stain is depending on phospholipase activity on a lot of plates, and this feature can be lost due to mutations in a central regulator, and that has been published by Lauren with colleagues from our institute. In the meanwhile, there has been more than 70 of isolates identified from a huge database that show this loss. And all of these isolates that don't have this very clear chromogenic phenotype on the agar would be lost in analysis. So we wouldn't address them as Listeria monocytogenes, although they are there, and although that these isolates are fully virulent. That was shown in a paper already by Norwegian group a couple of years ago. So when we're talking about theoretical framework in microbial analysis, we still have the target enrichment step, target isolation, and the target confirmation step, and even molecular tools that have been developed in the recent years to speed up the analysis. Need enrichments nowadays, but enrichment is a very difficult ride where you would like to give a chance to the pathogen, to the target organism, to suppress the commensal flora, but you wouldn't like to damage your target. So this is a very tiny way through it. And although a lot of research was done on the target isolation and the target confirmation level, where we see a very little advancement what regards enrichment. We have started this triangle in our theoretical framework, and I would like to show you and share some of the data with you. What we see usually in a broth is that we have a growing population of cells. Panos was showing this is in the slides. But we also have a condition, what we call this viable but not culturable state. I can jump over this because Panos was mentioning again that it's a very interesting population of cells. And we have currently started here, Patrick Mester, a professor in our institutes, has started an own lab on this, what we call these bacterial strains that are in a dormant state. Where the metabolic activities are somehow reduced. So you can't culture them on a plate, you can see them on plate, but they can cause fermentation problems. They can, to some extent, produce toxins. They can stay and retain a virulent state. So there is a lot of research now going on to define these intermediate physiological states to make risk assessments more targeted in that regard. So when we talking to food industry, and this is what we did a lot in FoodSafeR, because we had a vast sampling approaches out there. What is a pathogenic species? That's not clear for the food industry, especially talk to Listeria. We have apathogenic and we have pathogenic species. What can be addressed? Listeria monocytogenes is clearly a pathogenic species, but what is, for example, the indication when you have an apathogenic species being isolated? How are they transmitted through the food business operation? How can we clean them away? Are there strategies to more targeted cleaning and disinfection procedures? When are we safe if there's a contamination? What we see in a lot of cases, to what conditions can we say that a production system is safe again? And the final question is always, what is the price of a methodology? And we see a huge competition in the market for private analysis. And meanwhile, you can get a Listeria test for 12 euros. And that that tells you that there's a huge competition for which test to use. This is the usual procedure that we see. We have a sampling, swabs, we collect metadata on the sample. We have some environmental sampling to some dilution effects. We have enrichment, usually 1 or 2 step enrichment. And then finally, we can take that samples and put it into the confirmation steps. What has been not very much developed is the sampling, where to sample, in which frequency to sample, and by what technology to sample. This has been not much research being done on that. Usually swabs are used, but you can use little swabs. You can swab very little, tiny areas, small areas. You can use big swabs that cover far bigger areas there. And one project you have seen yesterday, the presentation of Lauren already, and Felix was this study for persistent European food business operations with 3 different countries that have participated. You have seen the data yesterday, but I think what was really a prerequisite is that we have standardized the methods that have been used in these 3 countries, because otherwise you would never come up with a comparable set of data. And I think Lauren has shown this yesterday, that a third of our scenarios, contamination scenarios there, were of persistent state. So sampling is much fun. You see here Felix doing the sampling approach, but how to sample? And it can be really critically biased. And this is what I would like to show you from that data here. Our consortium, the micro consortium that has worked in FoodSafeR together, has already collaborated in a previous EU project called PROMISE. And here we were asking food producers whether they know that they have been contaminated or uncontaminated in their food processing environment. And although they said, this one, for example, he has never seen any Listeria in the food processing environment, you can see if you do a very thorough sampling that at least you can find Listeria there. And this was the case for all other member states that participated in the project, even in Ireland. Those plants that said it's certainly uncontaminated had a higher isolation rate than those that said, okay, I know that there is some contamination going on. So whenever you look for pathogen, it's basically a matter of sampling, whether you identify, you don't identify it, and it's a matter of frequency of sampling that you need. So you have to think about a sampling plan as a very, very important first step before you start any analysis. And sampling triggers the outcome. And this is a paper that we have published a couple of years ago, where we were looking into dairy. And in that dairy, we had different compartments, like the cooling chamber, the cutting area, the washing area. And we are doing a lot of drain sampling to get an information on the Listeria contamination status there. And you see, if you just sample the drain water here in the drain, and you do a microbiome analysis, then you see around 250 species, bacterial species being present in there in the drain water. But if you sample that drain biofilm, that usually is somehow sticking here to the wall of the drain, then you see another 29 species, and only 17 of those species are shared by the two compartments in the drain. So that means if you're just taking the drain water, you're only removing a part of the microbiome being there. And if Listeria, for example, is fixed here into the biofilm in the drain, then you would miss it if you don't do a sampling that also includes the biofilm in the drain. And if you have ever done drain sampling, you have to go to the floor. If you have to do it on 40, 50 drains in a food producing environment, then this is a lot of labor work. And the people are not wishing to do this in a repeated frequency. So where to sample, what to sample, these are all sampling spots that have been Listeria monocytogenes positive. It's utensils, it can be slicers, it can be conveyor belts, it can be drains, it can be mats, for example, or it can be other things like hoses, all practical cases. And you need to think about which spot to sample before you start to do any further downstream analysis. We have thought a lot about this and published a method how we can best to improve that. And we have replaced, for example, the swab-based analysis by a sticker-based analysis. This is a sticker made of paper that you can fix to a surface. You can fix it to surfaces that are not very frequently cleaned, that are forgotten in a way, or where the company doesn't want to clean to, and then you can leave the sticker there. And if you have then removed the sticker, you can put it into analysis. Bacteria are sticking to this surface, the paper surface. In there, also viruses do this. In that paper, we have done it with viruses. You then can put it into your vial, and then you can start, for example, your molecular analysis. We were trying to convince food industry to use this sticker, but they were very reluctant to use this because this gives them a permanent control on the cleaning efficiency in their plant, and this is what they don't want to do. They would like to take the swab by themselves, and by taking the sampling, also can steer the outcome of the analysis. Yeah, you see very nicely that there's a high linearity in depending of the concentration. In that time, it was a virus that we were using, and you can see that that sticker are really capable of keeping back these viruses and at a very high frequency, independent of the treatment of that sticker. So if you use a washing cycle in between or a disinfection cycles, whereas the swab, you lose information over time. Good. How many minutes do I have? Nothing? Huh? Okay, good. Then I would like to finish with one information that we were all thinking of. What would be an ideal analysis in a food item? And this would be that doesn't need any enrichment. And we have developed and patented a concept that it's called matrix lysis, and this helps us by solubilizing solid foodstuffs. You don't change the number of microbial cells. That's especially important if you do quantitative microbiology. These cells are then harvested by centrifugation. Here you can see one example. This comes from cheese. You can take 25 grams of cheese, whatever. You homogenize it. We have developed this solubilization buffer. And with 2 or 3 washing steps, you end up with the bacterial palette in here. And then you can quantify. You can put it on a plate, or you can put it directly in the PCR, whatever. We have been using this technology extensively on a lot of different food items in the meanwhile. And we have extended the way how to treat and solubilize the samples with a class of liquids that are called ionic liquids. They are very remarkable, highly tunable chemical compounds. In Austria, we have created in the meanwhile the biggest, one of the biggest libraries for ionic liquids in collaboration with a company, Bryonic, an Austrian company that has developed this analysis pathway. And we have synthesized more than 1,000 of these ionic liquids and have tested them on food items. And a lot of them have very nice properties with regard to antimicrobial, but they can very nicely be used for this matrix lysis activity. We are now testing them and using them also in practical terms. You can also, bionic liquids, use it for extraction of biomolecules, so they not only might be used as a solvent, you can use it to crack the cells, for example. This is a very nice patented procedure where in two-phase system, aqueous phase system, you use an ionic liquid, you enrich your target molecules, the DNA, for example, you can harvest the DNA and put it then on a quantitative PCR. So what I would like to tell you, and this is the take-home message, now we have, if we're talking about an analytics in microbiology, we shouldn't just think on isolating and confirming the bacteria that we find. We need to invest more brain into how can we get access to the target and how can we improve the sampling and food processing operations. You have seen the data from FoodSafeR, where around a third of the samples, how many was it, Lauren? A fourth of the samples were positive for Listeria monocytogenes. After 40 years of work with Listeria, and I don't know how many paper panels, 10,000 papers on Listeria for sure, we still have a situation that we have a huge contamination burden in food processing environments. We have an increasing numbers in listeriosis cases coming back to the recent EFSA data. So the problem is not solved, it's exacerbating to some extent. And I think a big question mark needs to be put on how we're taking samples and how we're finally taking the conclusions out of it. Thanks for listening to my talk. I will stop it by now, and I hope you have some questions. Thank you.
In the interest of time, we're 15 minutes over. So we have coffee now. Let's try to be back here at 10.
Good morning. Good morning. My name is Veronica Lattanzio. I'm researcher at the National Research Council of Italy, and one of the part of my job is to elaborate on performances and fitness for purpose of rapid tests. So the presentation today, very difficult to speak, by the way, after Rudy and Martin, but anyway, the presentation today is not about how fast we can go with rapid methods, because we have already seen beautiful examples in the previous presentation, but is a little bit of focus on how to evaluate performances of rapid testing in relation to the real context. Sorry, I missed the pointer. In relation to the real context in which we apply them. Why rapid testing matters? Because it is applied in very different contexts. In official control, rapid testing is used. It's used mainly as a screening tool to classify samples into compliant versus suspect, but not indeed to make final legal decisions, which are taken by applying confirmatory methods. But on the other hand, in real scenarios in industry and food business operation, rapid tests are also used to get quantitative estimates. That enables the operator to take quick decision on acceptance and rejection of raw materials, batch sorting, screening large sample numbers in routine monitoring with the ambition, if possible, to reduce cost. So what we can see in practice is that the same tool can serve different purposes and therefore must be evaluated accordingly. What makes a test fit for purpose? We have regulation in Europe that apply to different classes of contaminants. Here are two examples that apply to mycotoxins and pesticide, for instance. Basically, regulation pursued the overall goal to protect consumers' health. That means, in practice, avoid false negative results and not raising unnecessary alarms. That means, in practice, to minimize false positive or suspect results that trigger confirmatory analysis. These concepts are translating into practical parameters, who are the cutoff, the false negative rate, which is normally asked to be kept low at 5%, false suspect rate. Normally, in evaluating this kind of parameters, the evaluation precision profile of the method is embedded. In routine checks, beside complying with these criteria and beside having the objective of protecting consumers, we can have other needs that also are added: ability to take quick decision, cost efficiency, smart batch management, which can be equally important as meeting performance criteria. When we evaluated the fitness for purpose of rapid tests according to the regulation, the only one criterion is do not miss contaminated samples. That means avoid false negatives and therefore prioritize consumer safety. And another important concept is that according to this regulation for screening tests, we are not really asked to evaluate method performances over a wide concentration range, but really against the concentration of interest, which is called normally screening target concentration, for instance, for mycotoxins, that normally is the maximum permitted level, or a concentration of interest in routine monitoring. So this is an example of how results from method validation can look like. And here we compare, for instance, two completely different, also in terms of sophistication, analytical approach. We have lateral flow immunoassay and liquid chromatography high resolution mass spectrometry applied for screening. You can see that we can get different precision profile, different cutoff, different standard deviation, but the most important is that if we evaluate them according to the criteria set for screening test, they both achieve the objective. The objective is to correctly identify samples at the screening target concentration. So from the regulatory point of view, no matter how analytically sophisticated is your method, you have to comply with this unique criterion. Now, let's add another perspective. The method validation, according to guidelines which are normally internationally recognized, depends on the method itself and how the operator apply it in the lab. Sometimes, to showcase this, I choose a worst case scenario. In this case, we had very bad performances, let's say, when validated the screening test for deoxynivalenol non-wheat at 50% of the legal limit, which was one of the chosen target concentration. But then, according to method validation, this method is not fit for purpose. Then this idea came from my colleague Christoph von Holst. He tried to collect 10 years monitoring data from the whole Europe. And what we saw is that in practice, only a very small percentage of samples fall within this concentration level where the method performs not in a satisfactory way. So what does it mean if you look at the figures? It means that if an operator applies this in practice, he will never experience the bad performances of the method at this concentration level. This was the empirical observation. And now, and after Christoph ended up looking for a statistical elaboration to put this in statistical terms, he ended up concluding that the bias theorem is the approach that can combine, at least explore the interaction between method performances, which comes from method validation, and the real frequency of contamination. So in very, very simple words, because this concept implies also the introduction of the conditional probability. When we perform official control, we want to know what is the possibility that the test gives a suspect result when the sample is non-compliant. This parameter is called method sensitivity and is calculated from method performances obtained in the validation exercise. But if you remember the two graphs, the comparison of performances and frequency of contamination, we could also ask the question, if I get a suspect result, how likely is that my samples is really non-compliant? And from a statistical point of view, the answer to this question is given by the posterior probability, which is the probability that the sample is non-compliant when classified as a suspect positive sample. So the relation between the method sensitivity, which is this parameter, and the so-called posterior probability, that means the probability that a suspect result truly indicated non-compliant samples, is set by the bias theorem, which is this formula that combines, in very simple words, the method sensitivity, which comes from method validation exercise, the prior probability, which is the real, takes into account the real frequency of contamination, and the marginal probability, which is the overall probability to get a real contaminated sample when it is classified as suspect. When we calculate these parameters for this worst case scenario at 50% of the maximum legal limit, taking into account this particular situation of frequency of contamination, you can see that we get satisfactory results in terms of marginal probability and posterior probability, which is the probability of non-compliance after a positive result. So according to this elaboration, the method still appears as a fit for purpose. Indeed, the situation changes if the frequency of contamination is different from this picture. This example is just to show you that behind method validation and performance criteria, which are indeed the main criteria for official control, we can further elaborate about the fitness for purpose, trying to merge the method performances from the validation exercise and the real world scenario. Now let's add another layer of evaluation of fitness for purpose. We talk more and more about One Health approach, and in our research we are all asked to take into consideration and quantify the environmental impact of food safety measures that we put in place. There are several approaches to calculate the environmental impact of analytical method. We choose as a case study the application of the AGREE metrics, which are described in detail in this publication. And the idea comes indeed from one of our youngest colleagues that realized that in routine, when validating or performing routine analysis, she was going to throw away a lot of plastic, for instance, when running lateral flow immunoassay, or solvent when running instrumental methods. So she said, I want to have a kind of tool that enables me quickly to quantify either the compliance with analytical performances and the environmental impact of my analysis. So the idea was put in place in a very simple way. We are going to automate this approach, but for the moment it's a guided procedure over Excel. The operator is asked, first of all, to define the purpose of the method for quantitative analysis or screening. Then it is redirected to another Excel sheet in which the operator input all the data it has from the analytical validation study. And then there is another portion of the tool in which the operator is guided to input all the description of the sample preparation and analysis, energy requirement, according to the 12 steps of the AGREE score calculation. To run this prototype tool, we choose to compare different approaches for detection of glyphosate in wheat. So we compared different tools for the same purpose. purpose, determination of glyphosate as a quantitative determination or screening. But the most important is that beside the analytical sophistication, all those cases were equipped with a different validation data set. And so here it is how the results of the tool look like. You can input all the results of the method validation, and then it compares the results together against the internationally recognized criteria for validation, and gives you a green mark or red mark according to the compliance. And then by analyzing the full set of green warning and red marks, it suggests the purpose of your method. So you can confirm that this method, for instance, which has been fully validated and has suitable sensitivity, can be used for official control. This method, which is flow injection without chromatographic separation, comply with most of the criteria, but there are some missing, so is suitable for self-monitoring and not enough for official control. The lateral flow assay was validated as a screening test and was compliant with the few requirements, so suitable for self-monitoring. After that, there is a third part of the tool in which it calculates the degree score by analyzing all the information that the operator input concerning the sample preparation, use of solvent, energy, and so on. And as you can see, as expected, but here you have a quantified vision, instrumental methods are less green than lateral flow immunoassay. And what was very interesting for us, because we were testing the possibility to fully automate the operation, also including sample extraction and analysis, is greener. What is nice of this kind of approach that if you look at the detail of the different steps, you can also have some indication which kind of step of your sample preparation and analysis you can improve to improve the overall greenness of the method. Indeed, there are other considerations to be made to choose the most fit for your purpose. But before concluding, let me add just the very last consideration about method fitness for purpose that somehow is linked to what we have seen in the previous presentation. I recently joined the strategic advisory group of the European Committee for Standardization on resilience of agri-food system. So it is a discussion about which standard and technical norm can support the resilience of food system in crisis and shock. So my responsibility there was to keep an eye on food safety standard, which is because the question was food safety determination, food safety control is business as usual also in case of crisis, or there is something that we can do to support the resilience in case of crisis. So actually the recommendation which is under evaluation by the committee is that indeed food safety can be something more than business as usual in case of crisis, especially for those contaminants who are impacted in case of shocks, and especially if we think about multiplexing. We have within the committee few multi-hazard standard method. We have one for mycotoxins, one for allergen, a couple, I think, for as completely not targeted method. So the conclusion is that indeed probably it's worth to invest more in standardization of broad scope screening tests to support resilience of food safety of food system in case of shocks, because they anyway can address the different dimension of resilience, enable broad situational awareness, allow rapid prioritization of confirmatory testing. So speed over precision without losing credibility. This should be a goal in case of standardization of the multi-contaminant screening method, because this standard compress time to signal in emergency. So this was the last layer of complexity in evaluation of fitness for purpose of a rapid test, which is if we think about real scenario, which is complementary, let's say, to the official control that has very, very strict requirement, no, let's say, space for discussion. option, no? And for trade-off. But if we think about the whole real scenario, we need to balance compliance with performance criteria, but also purpose and sustainability. So in the end, there is no universally best method, but only methods that are fit for a specific purpose and context. So the criteria against we evaluate them needs to be chosen according to the real need. Say that I thank you for your attention.
Thank you. I don't think we have time for questions. I'm sorry. Next up is Itz.
Welcome, everybody. As we've all been in the Netherlands now for a few days, you must know Dutch people love one thing: cycling. And if I speak for myself, in order to cycle, I only need one type of fuel: pasta. Pasta made from cereals. But here we run into a problem, because especially with fast mechanical harvesting, weeds that grow in between the cereal fields and contain a high level of toxic tropane alkaloids are easily co-harvested and therefore end up in your cereal product, such as your pasta. As these tropane alkaloids are strictly regulated and highly toxic for human consumption, they are strictly regulated at, for example, 10 micrograms per kilograms in buckwheat cereals. And although they can be successfully analyzed using a traditional sample-to-laboratory approach using liquid chromatography tandem mass spectrometry, this can be relatively expensive and is therefore not always available. Therefore, my aim within FoodSafeR was the development of innovative screening and quantification methods for the detection of these tropane alkaloids, which we develop in our laboratory and then to be used in industry. But to do this is not an easy route. No, crucial challenges need to be addressed. Firstly, your method needs to be both sensitive and selective to be able to detect those tropane alkaloids at extremely low levels in complex food matrices. Then it needs to be cost effective, as otherwise nobody is going to use it. And finally, it needs to be accepted by the eventual end users. Well, today I will take you through one of my projects within FoodSafeR, where we addressed all these issues by combining paper microfluidics with innovative immunoassay and mass spectrometry detection, and finally evaluating the usability of our device in industry in a user validation. Well, how does this workflow for rapid tropane alkaloid detection go? We start with the rapid paper-based screening, where we combine an extraction with a newly developed paper-immobilized liquid phase microextraction, PILPME, in which we enrich our tropane alkaloids on a small piece of paper, after which we can analyze this piece of paper using an indirect competitive lateral flow immunoassay. If you then get a positive screening result, you can transport that piece of paper towards the laboratory, where you can analyze it using both paper spray mass spectrometry as well as liquid chromatography mass spectrometry. Well, today I will mainly focus on the on-site screening part. However, if you're interested in the mass spectrometry part, which I also like a lot, please come up to me after the during the break, or when you have any questions, ask. But as time is limited, let's directly dive into it. Starting with the solid liquid extraction. Because tropane alkaloids are normally always extracted using water and formic acid, or water and formic acid and a bit of methanol. However, if you do this, you co-extract so much matrix, or as I would like to say in cycling terms, so much dirt, that you cannot run your lateral flow immunoassay with this extract anymore, or you really need to dilute your sample, thereby losing a lot of sensitivity. Therefore, we wanted to use a different approach, for which I wanted to use the pH-dependent polarity. And this is because all tropane alkaloids have a similar generic structure. And this indicates that the nitrogen atom on the tropane ring can be protonated under acidic conditions, making the molecule polar, whereas under alkaline conditions, the nitrogen atom is in its neutral state, making the molecule apolar. Well, what we then do during the extraction, we add a little bit of base to the cereals, deprotonating the tropane alkaloids which are present, making them apolar, and then add our organic solution. The apolar tropane alkaloids have a very high affinity for the organic solution, resulting in decent extraction recoveries, ranging between 70 to 80%. But now you may ask the question, can we run our lateral flow immunoassay directly with this organic solution? And then sadly, I need to say no. Therefore, we need to find a way to transfer the tropane alkaloids from the organic solution towards aqueous running buffer, preferably concentrating while doing so. Well, for this, we invented the paper immobilized liquid phase microextraction. How does this work? Well, you have a small piece of paper. This can just be regular paper. And you do a drop of water and formic acid on top of the paper. Then you dip the paper inside the organic solution. And because there's a difference in capillary pressure, your acidic aqueous solution will stay in the paper, whereas your organic solution will remain outside of the paper. Tropane alkaloids that come in contact with the paper become protonated, become polar, and stick on your paper. As there's a phase ratio of 1 to 180, this will result in significant concentration. Well, this is theory. Let's take a look how it looks in practice. We 3D printed a paper frame in which we can insert the paper from the top, ensuring both the constant positioning of the paper during the extraction and minimizing potential contact between the end user and the paper, which, in case of a positive sample, of course, will contain a high level of toxins in the end of your extraction. Well, we can move the paper frame on top of the extraction tube, and when the extraction is then complete, we can move it towards the elution holder, which contains a small amount of aqueous running buffer. Once the elution is then complete, we can pull the plug, and the nice thing is, the hole at the bottom of the holder is so small that your running buffer will not flow out directly. No, due to surface tension, it will stay in, and only if you move your paper frame a few times up and down, you will press out your running buffer. Well, we've seen theory, we've seen how it looks in practice. Let's take a look at the results. What we see is that after an extraction of 90 minutes, we reach an extraction recovery of approximately 80%. As there's a phase ratio of a 1 to 180, this will result in an enrichment factor on paper of about a factor 140. But now you might say, yeah, a very, very high concentration factor, but 90 minutes takes also very long. Well, this is true, but luckily, for atropine detection in buckwheat cereals, only a 20-minute extraction is already enough for screening at regulatory levels. Well, now we've covered the sample prep, let's take a look at the lateral flow immunoassay screening. Here, we again did some 3D printing. So I 3D printed a lateral flow immunoassay cassette, to which we can connect our elution holder. Then we can let the lateral flow immunoassay develop, after which we can measure the intensity of the test and control line using a colorimetric reader. Once we developed this, I bought 21 different buckwheat cereal samples. I first analyzed them with LC-MS to ensure they were blank, and once I knew they were blank, I either kept them blank or spiked them at 10 ppb, the regulatory limit, after which I analyzed them over 3 consecutive days. And what we here see, and then on the y-axis you see the T/C ratio, which means the ratio between the test line and control line, we see a clear differentiation between the blank buckwheat cereal samples and the contaminated ones. I then used a one-sided t-test to calculate a cutoff value at which there was approximately an estimated 1% false negative rate, resulting in an estimated false positive rate of only 0.7%. But now I already hear some of you think, yeah, of course you can do this workflow accurately. You developed it, right? And your colleagues, they are all experienced, but my quality control employee in industry, would they also be able to do this? Well, we had the same question. So as a next step, we wanted to go for user validation, where we not only wanted to evaluate our device on analytical performance, where we assess, okay, if we have contaminated buckwheat cereal samples, the quality control employee does the analysis, do we get a positive lateral flow immunoassay result? But also assess, are they capable to interpret this correctly? And finally, is it usable, where we assess both the ease of use as well as the workload. Well, once we had this ID, I converted the prototype we had into a real 3D printed biosensing toolkit with much enhanced its user friendliness. Work I did together with two colleagues, Lisa Portier and Erik Bij. And then, in collaboration with Michele Suman and Eleonora Rollo, we went to world's largest and by far my favorite pasta producer, Barilla, where we went from pasta production plant to pasta production plant, letting the actual quality control employees test our device. Well, what we see is that also here we again obtain a clear differentiation between the blank buckwheat cereal samples and the contaminated ones. Furthermore, if we look at the readout accuracy, and for this it's very important to realize we are looking at small molecules. So our lateral flow immunoassay principle is a competitive one, where the most intense test line is obtained for a negative sample and lighter test lines are obtained for positive samples. What we see, if the quality control employees analyze this with visual readout, there are quite some misclassifications. But if we align this with literature, this is quite expected because it's the other way around, as people are used to, right, in the corona test or the pregnancy test. Luckily, we also used a digital reader, where then we obtain a perfect classification, really highlighting the importance for this digital reader, but with digital reader also showing the applicability of the workflow. Well, then finally, we assessed usability, where we saw that our system obtained an excellent system usability score of 88 out of 100 and only a medium subjective workload, really highlighting that this paper-based workflow developed within FoodSafeR can be used for atropine detection in buckwheat cereals and ensure my pasta is safe for the next ride. Well, with this, I would like to thank some people. So first of all, my supervisor team, Gert Saantijn, Hans Elf, Twan de Bouvet. Then from Wageningen Food Safety Research, Erik Bij, Linda Wilmsen, Lisa Portier, Patrick Mulder, Jeroen Peters and the whole biosensor bioassay team. Laboratory of Organic Chemistry, Sipke Bosma, Seu Wang, Frank Klaassen. And then finally, of course, from Barilla, Michele and Eleonora Rollo. And all project partners. With this, I would like to end the presentation and I hope there are some questions, so feel free to ask.
Thank you, super presentation. Can you use it also on the field, so before you harvest it, so by farmers?
I would say one thing which is very important with tropane alkaloid detection is that you have a well homogenized sample. So we really looked at milled buckwheat cereals, which are then by definition, we assume very well homogenized. Yeah, and this is really the risk if you give it to farmers. I think the accuracy of the test will stay approximately the same, but you do really need to have the homogenized sample. And if you don't have it, well, your analysis results might be a bit biased.
Okay, Marco?
So, good morning all. It's my pleasure to present our work in task 2.3 from HoliFood. And it's about untargeted analysis by LC-HRMS of contaminants with the use of AI. So a lot of text there. And this is what was written down in the project proposal. So we've taken this directly, but this is short version. Deep learning for feature detection in LC-HRMS. Data tools for anomaly screening and marker identification will be developed. So pretty clear, I think. So, okay, so we did it together with BFR, our partner, and WFSR. And I first want to explain a few of the words which are stated here, and we heard them the last few days already a lot. But first I want to explain LC-HRMS again, and I want to use making coffee as an example. So this is not a coffee course, but maybe you learn a little bit about it. Coffee is like doing chromatography. So what you do, you put your sample on top of a column and you start to do an extraction. So if you use a little bit of water, so the first eluting, they will be a little bit acidic. If you use a little bit more water, then you get the sugary compounds, and if you even more water, you get the bitter compounds. So at a certain time, they will elute from the column. And all these compounds have a different structure and also a different mass. And you can already see that I depicted the mass by 4 decimals. If we use HRMS, we can determine this mass that exactly. So it's that accurate. So it's in the 4 decimals, so that's the difference of electron. We can even fragment them and then get a better understanding of the structure. And on the right, you see something what we call the chromatogram. So peaks are eluting at a certain time from the column are analyzed by the mass spectrometer. So then you can think, what's the big deal if you do targeted analysis? If you look for the coffee example, there are, maybe, I ask a good source, ChatGPT, 2,000 compounds, but there are also some volatiles. If you go to maize, there are approximately 20,000 compounds, and if you go to meat, 40,000 compounds. The last 2 examples were used by BFR and our institute to go and look if we can detect them in another way than we normally do. So here I already explained, the data files will be complex, they will contain a lot of compounds. We don't know what to expect, so there's no ground truth. And again, thousands of chemicals. So the chemicals, what we detect in a peak, we call them features in this case. So during this project, I learned different languages. I'm from the analytical chemistry, and I collaborated a lot with the people sitting over there from the AI, and we learned each other's language, I think, a bit more now, after 4 years. So a feature in our case with chemicals is the intensity and masses that relate to the compound we detect. And we put that in a feature table, like shown here. So a feature table is depicted like this. So we have a mass, we have a retention time, and then the intensity in one sample. These tables will grow enormously, but that's the input for our study further. So what's an anomaly? I think most of you have a kind of a picture of it. So if a normal population, one person stands out, you all sit here, and I'm standing, so I'm the anomaly here. So that's the same with samples. If you detect an anomaly, we can go in to the chromatogram and in the mass spectrum and find the structure related and identify the compound. Then something about deep learning. This is a typical workflow. So you have your data input, in our case, the feature table. We do some pre-processing, so we clean it, we remove some noise and unwanted compounds or other things which we think are not belonging to the feature table. Assess it by AI tools, different tools. Then we do a prediction, see if the outcome fits what we expected in the training phase, and go back. So this process is typical when you do deep learning algorithms. I want to focus a bit on the pre-processing first and then move to the machine learning algorithms. So let's start with the pre-processing. We developed a very nice pipeline. It's an open source pipeline. It uses existing tools, but also partly newly developed. And first, it will extract the features. So I have my HRMS data file. I can use this, and then I have the first features. Then I can combine them over the different files, remove features which we don't like it. Then we do some peak cleaning with an AI-based tool. We do alignment between the samples and between the days if you have multiple experiments, and also correct for intensity drift if the machine is a bit more sensitive on one day or another day. We also used visualization tools to characterize the data. So in this case, This is an S&E plot that shows you a bit from which samples are comparable. So if I have, like in this example, maize samples over the days, they should cluster together. But if I have spike samples, they should be separated from the rest, or QC samples. So that gives you a first idea on how well it performs. And also we have a multimodal anomaly detector that also gives you a better insight in your data. And we visualize it using an upset plot. For the ones who don't know it, it's like a Venn diagram that you overlay your different data sets and you get a better understanding on what's common and what's separated. Then to the AI. So we built a predictive AI model where we have as input the feature table. The feature table is then compressed and then further processed in latent space, but it's also built back to the original feature table with a reconstructed, so you can compare the outcome if the models don't do crazy things. Probably you all have used ChatGPT and that it hallucinates. We don't want that, of course, here. Then there is a branch that do the prediction of the samples. So if we have a blank sample or a contaminated sample, it will predict the outcome. We also want to understand it. You can hear it in each slide. I'm almost saying we try to understand it, visualize it. In this case, we use a common technique called Shapley that explains which features are important for the model. And that relates again to the exact mass. So to some data. So we built a data set. It contained chicken. We also spiked the chicken at 0.1 and 1 times the MRL. We spiked it with different compounds. So it was a very diverse set, also blanked. And then this set was split in different sets to train and to validate the model. And after the model building, so the iteration process I showed earlier, we have another set and try to see how it looks. Then we get this confusion matrix, which you see over here. And actually, it already looked pretty good. When Andrea was presenting it to me, I was like, oh, that looks good. But then he explained, I'm not happy. There is some day effect in the performance you cannot see in the confusion table. We have 2 false negative, 5 false positive, so it needs to be improved. And AI is quite data hungry, so we needed more data. So we could not process more data, but we had a big data set lying of urine samples of cow. So cow, of course, is something different, but for training a model on LCHMS data, a kind of work. So we have 1,200 samples. It contained samples from incured animals, it contained blank samples, contained spiked samples, and the model was trained again. Then we took the same data set as before, so the chicken samples, and it was put in the model. Then we got this as output. So back to Andrea again, I thought, not happy, Andrea, but then he explained to me again, yeah, that's what you can expect because a cow is not a chicken and it has never seen a chicken before, so you need to do something more. So to the next slide. So we did the pre-training now with the urine samples, and then we trained the model further and fine-tuned it using the chicken again. We put a bit more effort in the feature selection, so we pushed it more to the interesting signals because, of course, like Rudy explained, LCHMS contain a lot of different features, a lot of different masses. So we use a library which we have with a lot of compounds. We compare to blank, we do some normalization. Then we did the pre-training again with the pre-trained model to say it in a nice way. So we tried it again, and then this is the confusion matrix. And I thought it looked pretty good. So 23 were predicted as uncontaminated, 1 as a false positive, which I think is good to have a false positive rather than a false negative. And 26 were predicted as positive. So that was quite successful at that moment. So happy, it works with an AI model, with LCHMS data, with data transfer. And we can also explain it using Shapley analysis. So here you can see a bit the masses and retention time, so you can go back to your data to see what is causing the difference in the data set. And then we said, okay, if this works, let's go a step further and ask our colleagues from BFR, can we have a data set from you, which they processed with their pipeline, and also injected it in our model. So the model which was trained on urine, on chicken data. In this case, we injected it with maize data. Then it becomes more difficult This was the confusion matrix. So I discussed it with the colleague Jacob and Senia from Hey, what's this? How can I explain this? And then we had a nice chat because they were quite happy. And I said, how can you estimate if it's good? If you have contaminants like mycotoxins, they're always there almost. So they can be high, they can be medium, they can be low, but they're scattered over the samples. So they did another way of plotting, a cluster analysis. These are the highly contaminated, and these are the low and something in between. And if you connect this one to this picture, then it starts to make sense. Okay, the model picked up the really high contaminated, and the medium contaminated are a bit in this part, and the non-contaminated low are in this part. So that also worked, but a binary classification for this type of samples is not that easy. So this was the pipeline we developed. So we have our two matrices, the chicken and the maize samples. We have LCH-RMS. We have a pre-processing data pipeline, which is open source. We have the features extraction. We have the models in place, and also an outcome. So that's, in short, what we developed. So some conclusions and takeaways. We successfully applied transfer learning, so from the urine to the chicken to the maize. What the model output is meaningful. We did validation on the model, but we really like to validate it on more samples. So in the coming years, we want to connect it to our routine analysis and see what the outcome is. So next to our targeted analysis. But the take-home methods, we can detect anomalies using a tool like this. To conclude, I'd like to thank the people involved, Anne, Melissa, Andrea, Jacob, Senia, and Stefan. Two of them will have a poster presentation later today, so you can address questions to them as well, or go to the lunch meeting or the coffee meeting and drink a cup of coffee with them and ask questions. So thank you all.
First of all, thank you very much for this very nice presentation. I wanted to ask you if you can assess how far from the practical application of this methodology you are now as a group.
Yeah, that's an how far? We could apply it straight away. We analyze a lot of samples untargeted, so I would like to inject these samples in this model, but don't make the real decision. We still need our targeted approach next to it. How good it is, I think that's the answer. And I don't know yet. So that's something we can tell next year. And also we need more. Now we use a library. It would be nice if we have a foundation model that can determine each peak and see what it is and really tell me, okay, that's contaminant instead of using libraries. But we are close, and it goes fast. Because when we started this project, that was 5 years ago, in 21, I was not even aware of something like called ChatGPT. So it goes really, really fast. Yeah.
Thank you.
Okay.
Yeah, thanks for a nice talk. Just wondering for the matrix, right? So because we also talked about the garbage.
The garbage,
yeah. 95% garbage or potential useful data. There in infrared spectroscopy, right? There is only the way of so far the state of the art only allows for indirect determination, right? Mm-hmm. So you kind of conclude based on the infection, and the infection affects the gluten quality. You have a lower gluten content and so forth. So have you done any, or do you have in mind some particular experiments with different infection rates and also mycotoxin contents? Because that would be very interesting. We do the same thing with infrared spectroscopy. And talking about FoodSafeR and HoliFood and like collaboration conference, that could be an interesting subject to look utilizing your approach with FireS.
Yeah.
And with the infrared data into the matrix itself. But have you done anything like this?
The short answer will be no, as far as I'm aware. Yeah. But I think you're right. So if we can combine these results, so do first an assessment with infrared, then do the HRMS, and see if we can connect it, and maybe even some visualization, some pictures as well. Gives you more and better insight, and I think AI is so powerful that will really shine. So I agree. The short answer is no, but we should.
I mean, there have been undertakings like this before, but not. This was before AI and machine learning hype, right? So it's now the whole thing has to be reconsidered, and there's a lot of potential there.
And if you still have data, that can already be useful to start working on that. So that could be a nice chat later with a cup of coffee.
Good. So we will maintain our friendship.
And of course. Thank you.
Thank you, Marco. Next up is a dual presentation by Alexandra and Michele. I don't know who starts. Alexandra.
So, hello everybody. My name is Alexandra Schamann. I'm coming from Foxy, and I will share this presentation with Michele Suman coming from Barilla. So, I will start with giving you an insight into the fate of mycotoxins during the production of food, and then Michele will continue on the fate of furans during food processing. We could also say it in another way when we think of the dinner yesterday. I will guide you through the production of the pasta, and Michele will then add the sauce to it. So what happens with mycotoxins, which are the contamination in a raw material that is used for food processing? Are they stable or will they be degraded? These were the questions that we at Foxy asked ourselves together with Barilla. Since usually mycotoxins are analyzed in a targeted way using, for example, targeted like liquid chromatography, mass spectrometry. However, we have the problem that then the mycotoxins are can only be detected and quantified when they are in their original form, or at least in a form that is degraded, however, that is already described and also part of the method used. But when they are in a modified form that is not described yet, we cannot detect them, but they still could have negative impact on human health. So the aim of our study was to identify so far unknown degradation products of known mycotoxins that are formed during food processing. And we focused on two types of food, on the one hand on gluten-free pasta, and on the other hand on whole milk cracker production. Gluten-free pasta made from maize flour was chosen since the market of gluten-free products is rapidly increasing at the moment, and it is forecasted to further increase in the next years. As most of you know, maize is often contaminated with mycotoxins, also aflatoxins and fumonisins, and this is the reason why we focused on these types of mycotoxins in the first experiment. Then we produced whole meal crackers using whole meal durum wheat flour, and we did that since usually whole meal flour has the tendency to contain a bit more mycotoxins than refined flour, since it also contains the rather higher contaminated whole. In this experiment, we analyzed the fusarium toxins, T2 and HD2 toxins, for which maximum levels have been implemented in the year 2024. So now I will guide you through the production of gluten-free pasta. What we did was that we spiked maize flour with aflatoxin B1 and fumonisin B1 and B2 standard. On the one hand, the natural toxin standard, and on the other hand, fully 13C labeled standards, as Rudi said already before. This maize flour was then used to produce pasta in a pilot plant, mimicking perfectly the production conditions of the industry at Barilla. So we added water to the maize flour, then we mixed the dough, we heated it for 12 minutes at 75 degrees Celsius, then we made a brief extrusion, and then the pasta was dried for 360 minutes at a total of 80 degrees Celsius before it was then cooked. After all these steps, samples were taken, and then the toxins were extracted from the samples, which were then analyzed in an untargeted way using high-resolution mass spectrometry, which was just explained. We did it on an Orbitrap instrument, and thanks to the spiking of non-labeled and fully 13C labeled standards, it was possible to detect two mass peaks in the dataset that you can see on the slide. And these two mass peaks differ exactly by the number of the carbon atoms that a possible degradation product has. And the filtering of these mass peaks was done using an in-house developed software, MetaExtract2, that Rudi mentioned already before. So we did not detect any degradation product of aflatoxin B1, which was not so surprising since the production conditions were not so extreme. We detected one possible degradation product of fumonisins. Here, all 34 carbon atoms of the fumonisins that were spiked were still in the chemical structure, and it was obviously formed during the drying step. However, it was detected at very low concentrations, and so it was not possible to describe it further. It did not fulfill our quality criteria, and in the end, we could not confirm it as a degradation product of fumonisins. In the second experiment, we produced wholemeal crackers, and this was done in a comparable way. So we started again with wholemeal durum wheat flour, which was spiked with T2 and H2 toxin, again unlabeled and fully 13C labeled. Then the further ingredients like water, oil, yeast were added. Again, the dough was mixed, and then we had a leaving step for 2 hours at 25 degrees Celsius before the crackers were shaped, and then the crackers were baked for 6 minutes at 195 degrees Celsius. Again, samples were taken after all these steps, and the sample procedure was, as I described already before. In the figure, you can see the results, so the T2 and the HD2 concentrations in the individual samples from the flour up to the crackers. And in dark blue, you can see the T2, and in light blue, the HD2 toxin levels after spiking only T2. So you can see that HD2 formed out of T2 after adding further ingredients like water and yeast, and the levels increased after the 2 hours of leaving. Except for that, we did not detect any degradation product of T2 or of HD2 toxin. However, it's necessary to say that it could be that further degradation products were formed, but that they were at levels below the limit of detection. And in addition, it has to be said that it could happen that further degradation products could be formed when the processing steps, like heating steps, are longer or at higher temperatures. With that being said, I would like to give the microphone to Michele.
Thank you, Alessandra. And so we are driving you slowly towards the lunch because we had crackers for aperitif, pasta, and with the pasta you have to put the sauce, usually. So if we consider that one problem different from this one, from natural toxins and the way we use design of experiments and combined with high-level experimental phase with the high-resolution mass spectrometry, we did the same, but in this case, working on processing contaminants, and particularly with furans. Furans are volatile compounds that are generally formed from different roots in the food, and they have been defined possibly carcinogenic to humans since 1995 from IARC, and there is still not an official regulation for that. And it's a general, let's say, scientific rule of thumb to consider at now a level of about 100 ppb as a sort of threshold of concern, even if not a real regulation is present. Furans can be formed, as I said, from different kind of roots, and particularly from multiple precursors. So from carbohydrates, ascorbic acid, polyunsaturated fatty acids, etc., and carotenoids. And I highlight carotenoids because we'll see later on why. And substantially, you have several roots, but also at the same time, contemporaneously, several factors that obviously in a food processing can influence the results in terms of profile of different furan molecules and also the total sum of them. Means the composition, the processing time and temperature, oxidation, pH, etc. And substantially, also the mitigation strategies that are under evaluation in the food industry are considering obviously the recipe for itself, but also the presence of specific precursors, like could be, as I said, the carotenoids or the sugars, etc., and also consider potential alternative treatment, particularly relevant if you think about shelf-stable food sausage product in closed jars that has to be stable from the microbial point of view for months or a year, let's say. And yeah, so let's start to see what we did in the FoodSafeR project. This is a first step that was looking to the benchmark, so monitoring the products around the market to see, firstly, that we had to concentrate the attention on the meat-based sauces much more, because you see that the overall levels of the just tomato-based is very, very low with respect to the threshold that I mentioned to you. So we decided to concentrate the attention on the meat-based sauces, and the reason is related particularly to the presence of fats in the matrix that are strictly precursor for the generation of the furans, as I mentioned to you before. Substantially, I'm going now to illustrate to you the results of the different design of experiments that we set at both pilot and also industrial processing level. And a first interesting result is that obviously if you change the technology for the stabilization of the product, you have that there's a significant difference when you apply the classic, let's say, autoclaving with respect to a different kind of technology for the stabilization, which is called hot filling. And there's a clear difference in terms of reduction of the total amount of furans that can be produced during the food processing. So a first indication was about the let's say, processing condition of the sterilization of the product. And another thing that we found is that with respect to that, the sugar content and also the pre-cooking of the meat, like you can do at home when you prepare your ragu, let's say, is something that does not affect so much the total content of furans. Then another point was about the jars dimension. And you will see later on, but I can already tell you that if you reduce the jar dimension respect to keep all the other parameters stable, there's a shorter effective heat load and a better heat dissipation in the treatment that is reflected, is mirrored in terms of reducing the generation of the furans in the product inside the jar. This is another important thing, in conjunction with different kind of recipes. And particularly this is relevant because you see that different meat-based recipes are connected with a different, let's say, fat-rich matrix. And in this sense, you have that the complexity of the meat recipe within the product influence also the total amount of furans that is generated. So recipe is relevant, particularly relevant, once you have set the thermal condition, the processing from the sterilization point of view. And in this sense, I would like to show you an example of a design of experiments, one of the we did in the pilot and industrial phases, in which we were varying the temperature of the sterilization, time temperature of the sterilization, the size of the jars, and the presence of carrots in the receipts. And with repeated experiments, we found that as you can see in the variable important plot of this design of experiments, carrots play a pivotal role. So the carotenoids presence in the recipe in these conditions seems to be extremely relevant, which was not so much clear to us until this kind of project. And yeah, I can finally show you some further experiments in which we were trying also to exploit other mitigation action, like the addition of vitamin E in order to have a sort of an antioxidant effect. But you can see here, for instance, that these are two examples of experiments for the main furan, 2-methyl, 3-methyl, and the SAM, in which practically there is no real effect of the addition of the vitamin itself, and also trying to reduce through the technology the oxygen in the headspace of the jar does not generate a significant effect in terms of mitigation of the generation of the furans inside the jars. Final experiment I would like to show you is about, let's say, the effect, again, of the carrots, the impact. You see here, for instance, the standard production of a bolognese sauce with carrots and without, and it's clear the effect of the reduction, particularly for the 2-methylfuran and the 3-methylfurans, independently from the position in the industrial lines in which the jars are analyzed. So we can conclude, starting from the bottom of these slides, that substantially we could have a little bit of playing with the fat variability, the composition of the fats in the recipe, but it affects a bit, let's say, the final results in terms of furan generation, while, yes, you can optimize at least at a certain level the sterilization condition, but obviously you have some kind of restriction related to the most important thing, which is ensuring the microbiological safety. And so, quite surprisingly, we found that the real game changer could be the addition or not of the carrots in the recipe, which can really determine a significant change in the overall generation of the furan, and it's something that, okay, it can have an impact from the sensory point of view, but it's relatively easy and sustainable intervention that can be done at the industrial level. And all these things you can find within the next immediate months into the life learning section of the Digital Hub of the FoodSafeR project, because we are preparing a sort of decision tree with guidelines for the food industry in order to look to all these aspects and these steps to mitigate the furan generation in this food category. And with this, I would like to thank you on behalf of me and Alexandra once more. Thank you.
Thank you very much. One more presentation before lunch. Questions can be during lunch. Luc.
Good morning, everyone. I'm Luc van Ooyen. I'm not a part of HoliFood or FoodSafeR. However, I worked on projects relatable to projects which occurred during the HoliFood project. So I'm going to present about Beyond the Plate from Food to Fasta, a metagenomic-based detection method for foodborne bacterial pathogens. And my colleagues at Wageningen Food Safety Research were part of the HoliFood consortia, where they also used metagenomics. So Beyond the Plate, what do we mean with that? Therefore, we first need to know what's the current method of detecting bacterial pathogens in food. So for a lot of different bacterial pathogens, it needs to be absent in 25 grams. So it means that very limited amount of bacteria need to have the ability to check these very low abundance pathogens. So therefore, often the primary enrichment is being deployed so that a small amount of bacteria can outgrow to higher amounts, which then is followed by selective culturing and identification, hence the plate with specific colony-forming units. However, this takes 24 hours to 5 days, and it's also very specific. So it means for every specific pathogen you're screening for, you need to use a different standard operating procedure. So then the idea was, can we have an untargeted method to detect these bacterial pathogens? So then an alternative approach was thought about, from food to Fasta. And what do I mean with this? So instead of looking what we find on our plate, we're going to directly see what we can find in the DNA derived of a food sample. So therefore we deployed metagenomics to detect pathogen-specific DNA. However, we don't have a standard operating procedure or any standards in metagenomic sequencing to detect foodborne bacterial pathogens. So instead of looking at a plate, we're going to directly look at the bacteria derived from a food sample. But before we have DNA from a food sample, we need to have the ability to extract DNA in an efficient manner, and also have the ability to detect DNA from a very low abundance of pathogens of a food sample from different food types. And therefore I looked at different DNA extraction methods with different lysis conditions. So different ways to break open the cells to get the DNA out of a cell in your sample. So first I started with spiking different food samples with different bacterial pathogens. And then I used a specific targeted method to detect if I had the ability to detect very low abundance of the specific bacterial pathogens in my samples. So here we have a qPCR results of spiked ground beef, where I spiked ground beef with different concentrations of bacterial pathogens. And here we can observe that if you use thermal lysis with enzymatic lysis or mechanical lysis, the CQ values are roughly similar. And if you use enzymatic lysis, we had in general a higher CQ value, so meaning that we have less available pathogenic derived DNA in our sample, which result in more non-detected values if you go to lower abundance of pathogens in a food sample. However, when we looked at the DNA derived from these different methods, we see that if we use thermal lysis, that all the DNA fragments are very, very tiny. So it's not suitable for certain downstream methods. If you use bead beating, it gets better. However, it's 6.8 kb, which is good enough for certain downstream methods, but the method I want to use requires even higher molecular weight DNA. And this is enzymatic lysis. So then the idea was, can we potentially improve enzymatic lysis? Because we need to have this high molecular weight DNA. So then we deployed metapolyzyme. So instead of relying on one enzyme to do all our lytic work for us, we deploy a cocktail of 6 different lytic enzymes. And indeed, if we compare solely lysozyme with metapolyzyme, we observe that if we use metapolyzyme, we have in generally lower CQ values, resulting in always a detection, even in the lower abundance of pathogens in food samples. So then we decided, okay, good, we have a nice DNA extraction method. However, we don't want to use qPCR, we want to use an untargeted sequencing-based method. So we use Oxford nanopore sequencing, followed with metabolic classifier to classify all the reads to determine what kind of microbes are present in a given sample. So now we have a Sankey plot with an overview of some of the bacteria taxa being present in ground beef. So we have some pseudomonas, some lactic acid bacteria, and also some other meat spoilage bacteria. So I was quite glad, okay, this seems to work. However, of all the sequence results, only a very tiny proportion is actually bacterial derived. So it means that you need to sample sequence a lot to get a very nice overview of the microbial community. And if you want to look for this rare abundant pathogen then you need to go like even further. So we want to get rid of the cow-derived DNA. So then I looked at different host depletion methods. So the first idea was perhaps you can separate bacterial cells from food cells by a density gradient. So here we have different densities layered on top of each other, overlaid with the food suspension. And after centrifugation, we took different subsamples to have different fractions separated on density, with the idea that in one density, only the bacteria will be present, and then in another density, the food-derived cells will be present. However, when we look at the CQ values, we see of all the pathogens that in generally, we always lose some DNA derived from the pathogens in the different fractions. And if you look at the general targets as a readout for the general amount of bacteria and host-derived DNA, we do see that in some fractions there is less host DNA compared to the control. However, it also affects the amount of bacterial-derived DNA. So we deemed this is not a suitable option to deplete our host material. So then we looked at a different method. So we just started with a regular DNA extraction, followed with an antibody targeting specific methylated CpG islands in eukaryotic DNA. So we fish out basically all the eukaryotic DNA out of sample, and hopefully we end up with a sample with only bacterial DNA. Well, we did not seem to lose that much bacterial DNA compared to the control. However, we also did not really seem to lose any of our host material. So also this method was not really a feasible method to deplete our host material from our sample. Then we looked at the third option, differential lysis. So the idea behind different lysis is that you start with a food suspension. You add some enzymes to softly lyse all your food-derived cells while maintaining the microbial cells. Then you break down all the cell-free available DNA, and then you continue with your regular DNA extraction. And indeed, if you compare the control towards a different lyse-treated sample, we see a big reduction of host-derived DNA, while having limited effect on the microbial population of DNA. So then we optimized it that we can even treat high amounts of samples. So we can now use 25 grams to differential lyse it. And then indeed also in higher abundances, only a very limited amount of host material is present. So then we did again metagenomics. We saw some nice Carnobacterium, also some other Lactobacillus species. But still, the majority of the reads were cow-derived. It's still an improvement of the 0.07% which you had earlier. So it's a 100-fold improvement. So this is very glad, but it's still not on the level where we want to be. So then I investigated a little bit further. Where is this cow-derived DNA coming from? So here we looked at the coverage of all the chromosomes. And then the chromosomal amount of DNA is very limited. So less than 20% of the cow was found back once in the DNA. However, the mitochondrial proportion of the DNA is found almost 300,000 times. So all those reads which we saw in the host depleted were mainly mitochondrial derived. And indeed, if we go back to our qPCR results, we see that mitochondrial DNA is highly abundant present after host depletion. This brings me to my current challenges of this project. Mitochondrial DNA is highly abundant, and trials with alternative lysis methods did not harbor any useful results. So if anyone knows a method to get rid of mitochondria without affecting bacteria, please let me know, because it could greatly benefit my project and other sequencing projects in the future. And also another challenge is if there's a high abundance of bacteria, then it also competes with sequencing reads for lower abundant bacteria. So this is also a challenge that if you have a very sterile product with some pathogenic bacteria on it, you will find it back. But if you have a product with a lot of commensal bacteria and some pathogens, it's way harder to find them through metagenomic sequencing. I've also been asked to present an additional slide for Wageningen food safety research, because we already had a talk about different emerging risks related to globalization or to changes in climate. And this could affect new transmission routes of pathogens on food. And to continue studying this in the future, Wageningen Food Safety Research is working on a biosafety level 3 laboratory. So feel free to contact Wageningen Food Safety Research if you want to do research on certain pathogenic bacteria requiring these kind of facilities. Thank you very much.
Any questions or suggestions for Luke? A lot of questions. We have time for two.
Thank you very much for the presentation. I was really not an expert of this, but I think Oxford Nanopore has a system that can basically select the reads that you want to sequence and discard the rest, and I was wondering.
All right, everyone, please find your seats. This is one of the last sessions, but holistic risk assessment chaired by Jean-Marie, who will also do the first presentation, 30 minutes. We got 4 presentations coming up, then another break, and then a final poster session presentations. And then drinks, of course. Jean-Marie, take it away.
Good afternoon, everyone. So yes, the talk is about holistic assessment for food safety management. Of course, it's a part of HoliFood project, and here I want really to emphasize that is teamwork. So we were all of us working on work package 3, and particularly you've got people from Newcastle University, they are there in the room, Wageningen University, DTU, which is a Danish Technical University, UMVB, Veterinary and Medical University of Budapest, and INRAE, where I work. In terms of introduction, and that will be a little bit different but complementary, hopefully, from what Panos presented this morning. What I want just to introduce again, it's how we decided to set up the work package 3 in HoliFood. So we consider holistic assessment, risk, food safety management, and all this part, our focus was decision making. And as a driver, I took climate change to illustrate what I'm going to present, because I don't have a lot of time. Where we come from, we wanted to assess food system. And by assessing food system, we thought, okay, we need to evaluate the effect of different elements and activities related to food production, process, distributions, storage, preparation, consumptions. And we wanted to analyze that, assess that from economic, safety, health, and environmental perspective. Okay, we did not want to stay, strictly speaking, on food safety. But we realize that when you have so many criteria, you may have some conflicting response if you focus on one. And I take the example of microbiological risk. For example, if your goal is to mitigate the microbiological risk, and you apply a more stringent thermal treatment, you will increase the energy demand, which is not very good in terms of environment. And as you heard this morning, is not even very good in terms of chemical risk either. Okay? So, if I switch now to risk, what about risk? Risk analysis provides policymakers with the information and evidence they need for effective and transparent decision making. Okay, but when you talk about risk, and Panos said that this morning, in the decision making you have variability and uncertainty as well. In terms of variability, that means that the decision that will emerge from your risk assessment is not going to be optimal for everyone. The decision can go too far for some people and not far enough for other people. I say people, it could be factories, it could be farmers, for all the people involved in the food system. Uncertainty is different. It's a lack of knowledge or a problem of modeling and etc. But uncertainty implies that we might make a non-optimal choice between we may expect one outcome but something else occur. That's what we had in mind when we started. In terms of food safety management, food safety is a science-based discipline. And when it's about risk-based food safety management, it's the evaluation of management options where we weight available options. And as it is quoted by the FAO and the WHO, we can at this stage adopt a cost-benefit analysis. Now about food safety and climate change. You know that food safety, the climate change affects agriculture, fisheries, livestock. And because of that, the entire food system is impacted by climate change. And again, it's the FAO said that, it's not HoliFood, actually it was more the starting point. it's important to ensure that the food supply chain and regulatory systems are better prepared to adapt to the growing climate change impacts on food safety. That it was more or less the starting point of our thinking. Just one slide to illustrate the challenge with climate change. If I take only the temperature rise, an increase in an ambient temperature has an effect on all aspects of the cold chain, from the initial chilling, freezing of food to transportation, storage, and retailing. Changes to the cold chain system will therefore be necessary as ambient temperature rise. Okay. However, this often implies an increase in the energy consumption of food refrigeration systems and an increase in CO2 emission, which contributes to climate change. So if you don't pay attention, you create a vicious circle. And we started in 2019 in my lab working in an EU project, Protect, and we were, at that stage, we were considering only food safety and environment, and only one supply chain, which was the dairy supply chain. And we realized that you cannot anymore consider food safety risk in isolation, thinking of mitigation strategies, double-check the consequence in terms of food safety, and just note that there is a consequence in environment. We want really to go for a win-win solution where both environmental impact is reduced and the food safety is reduced, the food safety risks are reduced as well. That was the starting point of the logic. And now in HoliFood project, we tackle 5 dimensions that I'm going to present and 3 supply chains. So overall, what was the objective of this work package 3? It was to develop holistic risk assessment methods and tools to support regulation in a changing global environment. So the idea was to consider food safety, so microbiological risk, chemical risk, but also health, nutrition, actually, environment, and economic. That makes 5 dimensions that I will come back in a minute on it. And we decided to validate this approach on 3 supply chains, poultry, cereals, legumes, to deliver, finally, a generic, proven, holistic risk assessment method and toolkit. So we organize ourselves with some consideration about risk-benefit assessment, about the framework, so that has been published now. And after we work on the assessment of the 3, that is almost finished. And we have developed the tool. We used as well a living lab approach to help us with these 2 tasks. So if I go I'm going to cover the 3 supply chain, maybe not too much in details, but to finish with the general toolkit that we have in mind. About my supply chain, is a DTU who was leader on this task, but other people contribute to that. So first of all, they had to identify potential risk, understand exactly the maize consumption, and so they took 3 countries, but you will see that the other as well: Denmark, Nordic countries, France, Western countries, Hungary, Eastern Europe countries. That has an impact in terms of climate change. You know, of course, the 3 countries are different. And they characterize the baseline and they built some scenario regarding climate change. In terms of baseline, they analyze aflatoxin, cadmium, Listeria monocytogenes, Salmonella, but also dietary fiber, vitamin A, and other vitamins of the group B. In aflatoxin, they made really a zoom in, and they developed farm to fork model For that, they use previous work from the group of documents in Ireland that was developed during the Protect project that I mentioned. So they cover pre-harvest, post-harvest storage, food processing, consumer, and they converted all the info in terms of DALI. So DALI was mentioned this morning. One DALI is 1 year in perfect life lost. So, you know, DALI is a measure of human burden, actually. It's negative. So if you get a lot of DALI, you have a lot of lost years. And they had as well, they use as well with Newcastle, they use Delphi probabilistic techniques to try to capture all the elements that they needed to develop this model. And the idea is that they want to support regulation by quantifying the potential health impact of climate change on human health. And of course, they know that uncertainty is part of the deal when you want to consider climate change future and the projection, etc. So they built different scenarios. Let's consider that it was the easy one because they took only nutrition, chemical, and microbiology. See next, the lentil supply chain. That's where things start to be more difficult. The idea was to consider two drivers, the climate change, and everything is built on prediction of 2050, but also the diet transition, where people are going to eat less meat and probably more leguminous. With that in mind, we build four scenarios in total, but the first scenario is a baseline scenario. It's what happened now in France and Hungary. Why France and Hungary? Because these two countries, they eat lentils and they produce lentil. In scenario one, it's what happens if we don't do anything specific. So it's more based on the FAO lentil crop model. Scenario two, you get this extra 10% consumption by importing lentil from Canada. Why Canada? Because at the moment in Europe, we import either from Canada or from India. So we choose Canada. So the market is that we are not producing all the lentils that we are consuming in Europe is not produced locally, very far from that actually. And the other scenario is a 10% consumption, as the scenario 2, but by extra farming. And now that's the list. Here you get the 5 dimensions. And what is important here to realize that behind one dimension, environmental impact, for example, you get here 5 descriptors or indicators with different units as well. Okay? So if I want actually to assess my lentil supply chain, I need finally 16 indicators to assess. And after, I need to try to get some result interpretation from that. And that what we have here is the beginning of the interpretation. It's maybe not yet finalized, but here in black you have the baseline scenario, and here you have the 16 indicators. In red, you have what will happen in 2050 based on the FAO model that we, of course, we use as an input to calculate ozone, eutrophication, etc., the price for the consumer, profit for the farmer, Bacillus cereus. For example, Listeria here disappeared because the risk for Listeria was really considered as so low that we didn't need to continue. And after we got the scenario 2, which is import from Canada, and scenario 3. And here, between the two, for example, gas emission, you can see, and global warming potential, you can see that scenario 2 is worse than scenario 3. For profit farmer, of course, you can see that it's different. That's the reason why we say that overall scenario 3 is better than scenario 2. Of course, we will continue analyzing that. But overall, it's important as well to realize that even if lentil looks positive as a crop, it's going to increase the risk in 2050. And we have a lot of uncertainties, so we need as well, when it's presented, it looks like we don't have uncertainty, but yes, we have. It's just that technically, in this graph, if I put uncertainty, it's a sort of nightmare. We do the same for poultry. The scenario were just a little bit different. We consider climate change and the baseline, which is now O, versus 2 climate scenarios based on the IPCC report. The 2.7, which was the one used by the lentil team, and the 4.4. And here, I don't know if you see, yes, you see well, I suppose. 5 dimensions and 20 indicators. It's even more complex because of the chemical and a lot of nutritional benefit. And even in terms of environmental impact, it was more detailed. On the opposite, the economical impact was less detailed. But anyway, here, if you can see something, sorry, it's a little bit small, but you have France, Denmark, and Hungary regarding poultry. In dotted black, the baseline. In orange, the prediction with an increase in 2.7 degrees, and in red, with an increase of 4.4 degrees. You can see difference between countries. One big difference is France versus Hungary. For example, you get this eutrophication, and that is linked to the source of energy and the storage of energy in the future in France and in Hungary. If you need more details, of course, we can come back on that, but that is interesting. That makes really a big difference as function of the scenario, 2.7 or 4.4. That will make a big difference. In terms of microbiological risk, you increase the temperature, that increase the overall burden, the DALI, and that is quite easy to guess when you analyze the trends. And you know that in summer, for example, you have more cases than in winter. And so if you check all the trends, you can guess what will happen in 2050. Of course, that will be continued to be analyzed, but this group, which is actually Wageningen and UVMB mainly, they started to do something very challenging in terms of communication, which is I convert everything in DALI or in money. And I say it's very challenging because, of course, you have a lot of uncertainty behind, but you may have as well some difficulties to explain that you transform, for example, environment in euros or safety in euros. Anyway, the data here are presented per 100 inhabitants, so the value doesn't look big, but they are relatively. So here, the baseline, you don't have anything because it's relatively to the baseline. So it's a change, so it's a delta. A change in terms of DALI or change in terms of euros. And in terms of money, you can see that you have a big difference between the two scenarios. I arrived here with what we wanted to achieve at the beginning. So when we started the project, we checked how to assess, which techniques to assess in a holistic way this food system and food supply chain. We realized that some people were using domain-oriented methods, like life cycle assessment for environment, cost-benefit or willingness to pay for economy, and some were more in a risk-benefit assessment for health, nutrition, and food safety. There were people who were just encouraging using radar plot and all these descriptive methods that we did actually, because that has the advantage of being very transparent. So in terms of communication to everyone, including to the consumer, it's easier. And after some were more ranking, using ranking methods to rank, for example, mitigation strategies, and some were more in mathematical optimization, which works if everything could be really, really translated in terms of quantitative values. So what we did, we are there now. So what we did, we started with this idea of what we found in the literature. We get plenty of lessons from our 3 case studies. We thought as well in our group how to move forward, and we arrived to that I'm going just to describe. We say, okay, first of all, you need to define the scope of the assessment. That's, not jumping in the calculation too quickly. So that means an engagement with stakeholders, define the motivation and all the scenarios, define properly the food systems that you want to tackle. As function of that, define and select the dimensions that you are interested in, and the impact indicators, like you saw, 16 or 20. And you need as well to map precisely the food system, because, when I say that, geographically, for example, France and Hungary, you don't have the same thing. So you need to be relatively precise. When that is done with stakeholders, you can move to make the calculations, so estimate impact indicators, aggregate or not as function of the scope, include the uncertainty, and finish by interpret and report the results. It's where we are now, as HoliFood is finishing, and Work Package 3 is then finishing. And with that, I want just to say thank you to you, but also thank you to all the people in the room who helped, because it's really a teamwork. Thank you.
Plenty of time for questions.
Thanks, really interesting. To which extent do your model refer to compensation action?
Yes, for example, you have in mind that if people are eating more lentils, they are eating less meat.
For example, in the poultry, where you said there will be climate change, and the climate change will drive the storage temperature, but there will be, and this translates into illness, but there will be compensation, because there will be much more energy being consumed to reduce the impact of storage temperature again. So not mandatorily it must lead to illness. It could be also to lead stressing other impacts, inflows. To which extent does what is on the compensation side happening is reflected by your models?
No, so far, I mean, maybe Christina, you want to develop that even a bit further. But even, when we talk about each dimension and each indicators, the setup was quite difficult. So we didn't go to this level of trying to add a sort of complexity. We didn't touch that. And I thought you were thinking of, for the lentil, we were thinking at the beginning, if people are eating more lentils, they are eating less meat. So in terms of DALI, maybe in terms of microbial risk, maybe it's better. But that makes the analysis, you double the analysis. So far, we just try to establish the methodology, and we are not yet at the level of having a very, very complex model. We will build step by step. Maybe for the maize, because they were considering only the health, so it was a little bit easier. But for us, we are not yet at this stage.
Thank
Thank you. Super.
Next up is Nina.
So yeah, me again. I also presented yesterday, but now I'll be presenting for work package 1.3, and we also looked at holistic assessment, and in this scenario we're using this outranking method, so different than what they were doing with the mathematical. And I'll be sharing our chemical hazard. So this slide is from our HoliFood sister project about what is an emerging food risk, because we're mostly looking at holistic assessment for emerging foods. And I think we've also discussed it yesterday. It can either be a new hazard or an existing hazard with an increased exposure or a change in susceptibility. So we also just heard, why do we need holistic assessment? I think Jean-Marie really explained very, very well. So I won't dive in deep again, but we want this broader view. We want to have a broader assessment. And in our assessment, we also include proactivity and a participatory approach. We are doing proactivity with drivers of change, and participatory, we're asking experts and stakeholders to join in, because we don't always have this data available, what we also saw in the example from Jean-Marie, I think. Our goal is decision support for food safety management of emerging risks with a broader perspective, or a holistic perspective. This is our framework. It's a little bit big, but we start at step 1. So first, we want to have a proactive approach for an emerging risk. We have our drivers of change that we identified, and Matthijs will talk about that a little bit more. And then we want to have an early warning. In step 2, we're looking at the holistic and participatory side. So for participatory, we have our stakeholders and our experts. And then for holistic, we're using triple bottom line, or also known as the 3 Ps, people, planet, profit, to capture that broader perspective. Then in step 3, we're doing our calculations, and this is based on a multi-criteria decision analysis, and specifically PROMT or PROMETHI, depending on how you pronounce it. We want to have our food safety management activities that are appropriate for our emerging risk, which we can get from our stakeholders, experts, and other sources. And we want to weight our criteria, again, with this participatory approach. Using the outranking method, we can then get our best evaluated food safety management activity package or set. I'll specifically demonstrate this framework with a case study on mycotoxins in New Zealand. And I first would like to thank our partners there. It's PHF Science, based in Christchurch, and they generously hosted me for 3 months, where I collected this data and I talked to all of their experts in their network. And why is mycotoxin emerging risk? I think yesterday we established that it's known risk for a long time. We also talked about how these drivers are influencing mycotoxin occurrence. We just heard it again. Climate change is a really important driver. And I think this picture, it's not of New Zealand, but it definitely demonstrates what's happening with the warming. So here, there is no increase in this model. And here they say, I think it's plus 5 degrees. And all of a sudden, you see mycotoxin occurring in places it didn't occur before. And this is also happening in places like New Zealand. So for our first step, we're taking a proactive approach. We're looking at drivers of change. We identified 6 imported drivers. And again, Matthijs is discussing the drivers in more detail, so I won't bore you with all the details here. But I think a lot of them are recognizable, especially for a country like New Zealand, which relies on import. You can imagine that geopolitical instability and supply chain can have enormous consequences. Step 2, the participatory approach. We've done 2 different methods. So we have our expert interviews, and we had a survey. And these expert interviews, they were really important at the beginning to understand what was going on, which activities to include, and how to weight these criteria. And then in the survey, we had a larger group of people who really looked at the activities and at the criteria at the same time. Again, a really large slide. But these are the criteria, and you can see the 3 Ps again. So we start with our people, so with 3 people criteria: health risk reduction, farmer work adoption, and social acceptance. 3 planet criteria: environmental impact, ecological footprint reduction, and climate future-proofing. So the ability to withstand future climate change. I think that's also a little bit resilience-oriented. And then for profit, we have fixed cost, variable cost, and feasibility. So can we actually implement? How much would it cost to implement? Then with our experts, we decided on 6 food safety management activities. There were more, but we got to a point that the survey would get too large, so we had to reduce. And we've also categorized them on impact scale. And these impacts, they're the primary scale, so they will still have impacts on other scales, but we had to make some choices. So the first food safety management activity is surveillance by industries, then maintain and improve lab capabilities, especially to analyze low-level contamination. In New Zealand, they're quite reliant on Australia's labs. Then for the local impact scale, we're looking at planting and crop timing and the fungicide management. For global scale plant breeding approaches, they have a little bit of a different view on genetic engineering as well, and also the increased awareness of mycotoxin risks, which I think in this room there is awareness, but in a lot of other places there is no awareness. Okay, then first result, here we have our triple bottom line, or our 3P, the weights, and this is in percentages. I realized later I didn't add it to the slides, but we have 18% health risk criteria, so it's the highest scored, most important criteria. And as you can see, all the costs are scored a little bit in the middle. I think people don't always know what to do with this environmental or economic side of things. And then they had to score the food safety management activities on these criteria, which is a difficult task. And from there, we have our Prompti outranking method. So for national impact, we can see that surveillance by companies was preferred. It has preference 1. Strategic crop timing is preferred, and plant breeding. So a decision support document that could be written would be, have an activities package that looks something like these top preferences. Of course, different stakeholders responded differently to the activities, but also to the weights. But as this is preparation for a publication, I decided to just show you the overall. But that could also give more insights into how to manage that package of activities. So maybe there is a stakeholder that has some negative impacts from a certain activity. You could incorporate a subsidy or a scheme to promote adoption. So our aim, decision support with a broader scope for emerging risk food safety management. I think we've managed to capture that and to demonstrate that with this chemical case study. Then the proactive approach is really about when to act and where to allocate resources. Participatory is who to consider. And the holistic is to capture that broader scope that Jean-Marie told us all about. And in future research, we would really like to show more cases with this framework. Thank you.
Yeah, thanks, Nina. My father suffers from color blindness. Unfortunately, I haven't inherited that. On slide 5, this is a very very often showed slide. Just wondering, is there something wrong with the color slide? Because shouldn't that actually indicate the expansion of aflatoxins towards the north?
I think it does.
Shouldn't that be red?
Maybe I chose the colorblind version.
Right, because it's moving like C is, I think, the plus 5 degrees Celsius scenario, which is actually indicating more aflatoxin, but according to the color code, that should be less, right? Sorry? It's for rice, okay.
Okay.
Okay, but would red not mean that there's an increase?
Yeah, but blue also means an increase. If you look, we start at white and we're moving towards.
Okay, so there is no red in. Okay.
In
so
this rice version, apparently no.
Okay, in the rice version. Okay, yeah. So that was the thing. So that's a new thing. And I was actually wondering, because you are the ones who will do this scenario analysis or prediction. Isn't there because Batilane, that's why I was confused, from 2016, right?
It's an example.
And so I'm just wondering, can we expect as a result of our joint efforts, maybe for 2030, that we can kind of move on from the Batilane to the next level of having a prediction for with a paper which was published in 2026 or 2027, because it's always referring to a paper which is 10 years old. That's what I mean. Is there?
I just really like this picture. I think there are a lot of
I like
papers.
it. It's just. I think with the dynamics of climate change, one can easily argue that when we show it at a conflict like this, so guys, where are the new, where's the new publication, and why are you still referring to the Batilane paper published 2016, and I think they have maybe worked 2014, 15 on that. Yeah,
I think it's great if we could have a publication like that.
Yeah, that's why I'm asking you whether you or some of the co-authors know about
no, I don't.
Endeavor to actualize these Batilane figures. Although I like Paola Batilane a lot, but
I have no knowledge of that, but maybe we could work on something. I just really like this picture, so that's why I included it.
Yeah. Okay. Thanks. That answers. Thanks.
He wants to see more red, so I wore the right jacket today. More questions? No. Want to invite Matthijs to the stage? You're away. Take an email.
Okay. So my name is indeed Matthijs. I'm a PhD researcher at Ghent University, and I'm affiliated with the FoodSafeR project. And I was asked today to provide you with a perspective, the FoodSafeR perspective on the drivers of change, which you have seen a lot of times popping up in multiple slides yesterday and today. Identifying those drivers of change was also a very important part of at the beginning of the FoodSafeR project, and also led to this report on drivers and key factors for the emergence of food safety hazards and associated risks. But first, over the days and all the presentations, I hear a lot of terminology: drivers, factors, stressors, trends, megatrends. They all are referring somewhat to the same or something different on different timescales, different levels of granulation, and so on. So it's very important to define them. But however, apart from the semantics and the terminology, I think the effect and the impact is also more important. So with FoodSafeR, within FoodSafeR, we chose for drivers and drivers of change, because we can say that our agri-food systems are under pressure, impacted by those drivers, like we already heard about climate change, geopolitical instabilities, population change, urbanization, depletion of natural resources, and much more. And because of those drivers, international organizations like the Food and Agriculture Organization call for action. Action to transform our food systems to more efficient, inclusive, resilient, and sustainable agri-food systems. But these drivers of change can also impact food safety. So when we're transforming food systems, it's also important to recognize food safety from design onwards. And I'm referring to design because, for example, these drivers are also impacting food businesses, and those food businesses innovate in response to those drivers of change. For example, innovations on how to produce more sustainably or adopting circular economy practices. And all these changes to the way how we produce food, how we consume food, maybe we need to rethink of food safety as well. This can be of known hazards, put some more focus on the hazards we already know, which was also the main focus of the FoodSafeR project. And we're speaking about re-emerging risks. We can also give attention to new hazards, emerging risks. But then we also have some sudden events, disruptions, which can be linked to those drivers of change. And then we don't define them as emerging risks, but we call them food safety shocks, events that lead to an increased presence or emergence of specific food safety hazards. So this is an overview of the complete FoodSafeR project. And as I said at the beginning, we started with defining drivers of change and identifying those drivers. As you can see on those on this figure where all the nice work packages, the input of those drivers was leading into case studies on microbiological food safety, which was presented yesterday by Cynthia, case studies on chemical food safety linked to the drivers of change presented this morning by Rudy, and then also on the influence on food businesses and how food businesses deal with those drivers of change within their food safety management system, which will also be presented by Lisbeth Jacksons later on. So, drivers of change, FAO has a very nice publication recently out, and drivers can actually be defined as macro-level factors, which can be categorized according to their social, technological, economic, environmental, or political influence. Together with the definition of emerging risks, like I already said, known hazards with a new or increased exposure or a change in host susceptibility, and then also that's re-emerging risks, and then also the new hazards, which have a significant exposure. And I'll add to this because EFSA excludes the sudden events, the sudden disruptions, so I also add to them the food safety shocks. And then drivers are defined or described by EFSA as some factors that may act as modifiers of effect on the onset of emerging risks, and that they can either amplify or attenuate the magnitude or frequency of risks arising from those various sources, like I said, steep categorization. So within FoodSafeR, we looked at all the drivers on our food system, and specifically how can those drivers impact food safety. And I also want to point again to this publication of the FoodSafeR team, which was published, which also links the hazard perspective on emerging food safety hazards and emerging risks together with the FoodSafeR Hub, which was also presented to you yesterday. Like I said, we had a definition of drivers, then we can continue with how we identified them. And within FoodSafeR, we focused on known hazards, established hazards within our European food system. And for the identification process, we looked at those known hazards, dived into literature, did a literature review to identify factors that are contributing to their potential emergence. Those factors were then grouped using a thematic analysis, and drivers were identified. We did some expert surveys, and also with our broad international advisory board, which was also already referred to multiple times, we organized workshops to further refine those drivers and also to validate this. Both approaches are also published by our colleagues at Wageningen and the Austrian Institute of Technology. The result of this was a set of validated drivers, 11 drivers categorized according to STEEP. So we have the social drivers, technological, economic, environmental, and political drivers, each with a set of sub-drivers, and I'll go a bit more in detail on two examples. First example, social driver of consumer behavior with four different sub-drivers. We have dietary choice, the individual choice of consumers, which can have an influence on food safety. We have the consumer knowledge on food safety, consumer awareness and attitude towards food safety, and then also the broad public awareness on food safety. Think, for example, about media attention. Another completely different example, which Nina also referred to already, is political driver of geopolitical instability with two sub-drivers, war and conflict, and fragmentation between nations. For war and conflict, we can refer to the recent conflict in the Middle East or to Ukraine, Russia. And for fragmentation between nations, we can refer to some trade problems. Think about Trump tariffs, if I can call them like this, or the Brexit. And for the continuation of this presentation, I will briefly show 4 examples of how we use those drivers of change in different work packages along the FoodSafeR project. As first, this was also part of the paper on identification by our colleagues at Wageningen. You see a very nice graph with all the categories of known hazards, and then at the bottom, the drivers of change. So they performed an expert survey with food safety experts to measure the association between different hazard categories of these known hazards and the drivers of change for their re-emergence. If you look at the bottom line, because in the next case study I'm going to focus on them, you see the very broad group of pathogenic bacteria, which are also all very highly associated with those drivers of change. So at Ghent University, we wanted to know, okay, what is behind those drivers of change if we look at very specific microbiological hazards, known microbiological hazards. And I will, within work package 2, I will focus on just an example of Listeria monocytogenes. So we went back to the initial literature review where we identified the drivers and looked what factors are now contributing to their re-emergence. And I will give a very simple example. If I zoom in on the driver of dietary choice, the social driver within consumer behavior, you can see that multiple factors that are established in scientific literature, which can maybe contribute to their potential re-emergence. And a well-known example, what we can all expect is, for example, the increased consumption of ready-to-eat foods with an extended shelf life under refrigeration, very well known for Listeria monocytogenes. In that same work package 2, based on those drivers, we also, multiple case studies were identified, which were described yesterday. So I'm referring back to the innovations now. So food businesses are innovating, or in our food system, there are innovations. And to those innovations, we need to maybe rethink food safety, or we need to gather food safety information. I'll show here 3 case studies that are already published now. The first one is on production, aquaponic farm production. If you just think from a very narrow food safety perspective, you're using aquaculture wastewater to irrigate your plants, you would say a very bad idea. But we're looking at a more holistic perspective, so we think, okay, maybe it's a good idea from a sustainability perspective, from applying circular economy principles. So how can we make this work in a safe way? Then we have spontaneous vegetable fermentations. It's a processing way. Cynthia already told, is this emerging? Well, in some countries it is very established. It's a traditional way of producing and preserving food, but in other countries and other markets it is indeed emerging, especially when not using starter culture, so doing it in a spontaneous way and not using pasteurization. It's okay to do, but how can we do it safely? And then we also focused on a case study on the distribution side. During COVID, we all started to consume our food a bit differently. Maybe you couldn't go to the restaurant, so you ordered your food online and it was delivered at your doorstep. A lot of news articles were out there. Is this safe? Is this hygienic? What is the contribution? So we thought, what is the contribution of this last mile delivery to food safety? And we also have a publication on this. All case studies and related publication to microbiological food safety in the FoodSafeR project are gathered in a special issue in the International Journal of Food Microbiology, Navigating Emerging Microbiological Risks in Novel and Alternative Food Networks. Again, you can innovate in your food system, but you should design them with food safety in mind. A third part, what we did with the drivers, or actually what our colleagues at Wageningen again did, was a categorization of the drivers that were identified. All those drivers were also using experts, were categorized via multi-criteria analysis based on their manageability and their negative health consequences. Manageability, we look at their controllability and their volatility, and negative health consequences, risk, we look at likelihood and the severity of the associated hazards. This is also published. I forgot to put the publication, but if you type in categorization of drivers, you will find the publication by Nina as first author. And then finally, I already said there's the perspective of food businesses who have their food safety management system. All those drivers which are present in the food system, for a food business, they're present in their external business environment. And on how they deal with this, I will give the floor in a minute to Professor Lisbeth Jacksons, because she will focus on how food businesses deal with those drivers in their food safety management system, and especially related to those food safety shocks, how they build resilience towards them. And I'm very happy to share that this was, as of this week, also published the first part of 2 publications in Food Research International. Thank you.
Thank you. Very impressive. When I was looking at your drivers, I was thinking perhaps taking even migration into it, or globalization, because I missed that a bit.
The migration, or?
Yeah, because there's so much going on over the world, not only politically, but also taking food, whatever.
Yeah,
and
travel and migration is actually one of the sub-drivers we indeed identified. So I didn't show them all because we have 39 sub-drivers, so I didn't want to go too much in detail. But if you look into the publication, you will find migration and travel as one of our sub-drivers indeed.
Okay, that's all right. I had the impression you mentioned them all.
Same with demographic changes, eh?
Yeah.
Yeah.
Yes, indeed.
Yeah, but behind this set of drivers, we also have sub-drivers, and this is one of the sub-drivers. Yes, we have 39 sub-drivers. Yeah.
No more questions? Perfect.
Thank you.
Okay. Yes.
Indeed. My name is Lisbeth Jacksons. I'm also from Ghent University, from the same Department of Food Technology, Safety and Health as Mathis. And I will continue with the story because we use those drivers also to reach out to food business operators and to see how these drivers are impacting them, and especially then their activities within their food safety management system. So this was a work done in the frame of work package 5 of the FoodSafeR projects, and it was also done in collaboration with Professor Pieter Loening from Wageningen University here. So those drivers of change that Mathis just explained, as said, they are impacting food businesses. So here we really look to the HACCP and the prerequisite programs that food businesses have in place in the frame of their food safety management system. So these drivers are belonging to what we call the external business environment. And actually they can influence food business in 2 ways, as Mathis already addressed a bit, via innovations, or they can also provoke a shock. And so innovations are changes done within a food business which are intentional. Think about a change in consumer behavior. New R&D will be done, new products will be developed and will be introduced. So these are things which are under control, which are well known and understood by businesses and are more the long-term strategies to respond to certain things which are changing in that external business environment. Food safety shocks, on the other hand, these are unwanted, often also unknown, or things which are happening suddenly. Companies are not prepared for that. And like that, companies can face food safety hazards that they are not aware of, or maybe don't have under control anymore. And there comes then the resilience principle in. So the resilience principle is a well-known principle which is used in many different businesses. And in the food world, let's say, resilience was first defined in the terms of food security, so to have enough food and to have food accessibility. So that was already in the 2015. Then Ine, with her group, started here to look to resilience of food chains. So how can a food chain react to certain shocks which are happening, and how can they recover from that? And so we are looking to resilience of individual food businesses. So the food safety management system of individual food businesses. Yes, so here you see, and we were thinking, okay, how could food businesses respond to that? How could they build a resilience strategy to anticipate on this and to deal with these food safety shocks? Yeah, and to narrow it a little bit down, because of course you can have chemical issues, physical hazards, whatever. So we focused also here on microbial food safety. So as a research framework to collaborate with food businesses, we used the DISPER model. So this is also used by other research fields. So this is to identify the drivers, to understand how are these drivers resulting in a pressure, and how is it then changing the state. So the state was here the microbial food safety status of the food businesses. Do they have more problems with pathogens in terms of raw materials and products, etc.? Then the next step is knowing the impact. So on which activities in a business does these pressures have an impact? So is it more on the monitoring? Is it more on the validation, verification activities? And then finally, okay, how could they respond to deal with those shocks? So this model was applied with food businesses. So we had several workshops for that. So a first workshop was dealing with identification of drivers, pressures, and impact. And the way we asked the questions, we also translated this in an online tool, which is available on the FoodSafeR Hub. I will show this immediately. So food businesses or researchers can use that tool to identify potential drivers. What are then the pressures and how is it impacting, or on which core elements of your food safety management system are these playing? And a second tool is then developed to capture responses. So responses on the first hand at an individual business level. So what can you do to be more responsive and to be more resilient as an individual food business? But we also identified some responses, again, more at chain level, so more from the food systems approach perspective. So we focus mainly with Belgian food processing companies to do these workshops. Why? Because they're all in the same legal environment and in the same region, so not to have too many variables which are playing around. Well, this is a little bit how the companies look like. So it's a typical snapshot of our Belgian food industry. We have a lot of family-owned businesses, which can be quite big, which are really very much export-oriented and which have all kinds of certificates. So let's say that they have a quite mature food safety management system already. So if we were asking out of these drivers and sub-drivers, what are the major drivers that you really feel every day? So legislation, policies and governments came on top. They said, okay, food legislation and changing food legislation, this puts really a lot of this is impacting us a lot. Together then on the third place, you have good practices and standards. So certification schemes which are changing, best practices which are changing, for instance, from international organizations which have new guidelines. This is really impacting a lot how we need to change our food safety management system. On the second place, it was climate change, and I'm more specific about temperature and extreme weather events. They really felt in the more unpredictable microbiology of many different raw materials. On the fourth place, we had technologies in food processing. So novel or innovative processing techniques which are out there and that they need to deal with. And then consumer behavior, and surely about consumer awareness and consumer attitudes is quite impacting their daily food safety management system activities. So if we look to the ranking in the previous work we did with experts, etc., legislation didn't pop up. Yes, and also if you look in the literature, a change in legislation and standards is not popping up. So while if you ask this to food businesses, it's the first thing that they say. This is putting a lot of pressure on them. So as said, the pressure is really about microbial food safety. So a sudden or an unpredicted change in microbial contamination, pathogens that they were not aware of and suddenly they have to deal with, or they get other prevalence level or other concentrations. And these are 5 major pressures that were identified. So changes in raw materials, the microbiology of the raw materials which is changing, differences in storage and distribution methods, and which is leading then to changes in microbiology, product design, if they need to change into products, need to change into recipes, they get another microbiology, another shelf life. Same with food processing technologies. And then finally also, of course, these stakeholder requirements. Think about the European legislation on listeria, which is recently changed. So this is really impacting a lot these food businesses. So then we were interesting, okay, these changes are occurring. Changes in raw materials, for instance, are driven by those drivers. Yes. How is it then impacting typical activities within a food safety management system? So therefore we used the diagnostic tool of Wageningen University, which is stating that the major activities in a food safety management system are on the one hand quality control activities such as preventive measures, interventions and having a monitoring system. And on the other hand, you have more quality assurance activities like validation, verification, documentation. So this is a kind of a heat map. So here you have all the drivers and to which activities are they impacting. And of course, the bigger the ball is, how bigger the impact score is. So typically for changes in raw materials are in fact impacting preventive measures, you can see. Also the monitoring system. And then also here, validation activities, for instance. This is a bit more clarified in this representation. So the food safety management system, you see there centrally with quality control and quality assurance activities. So if our shocks are influencing the microbial status of our raw materials, it has an impact on the one hand on preventive measures because they need to look for other suppliers, specifications need to be changed, maybe they need to look for other regions, maybe a new cultivation system need to be looked for. So these preventive measures linked to supplier selection and raw material selection will be very important. Also, they need to rethink on how to do the monitoring and the sampling plan and how to do the microbial analysis because working with the same analytical techniques maybe is not, as they did for all these years, is maybe not enough. They also need to rethink about their validation system, because of course if your microorganisms of your raw materials are differently, maybe your heating treatment needs to be differently, or pH or whatever, so they need to rethink and revalidate. So quite some impact of only a shock of changing raw material microbiology, which have then a huge impact on different aspects of that food safety management system. So this is just an example for raw materials, but we worked this out for all these 5 different pressures that were identified. And this is indeed published in the paper that Mathis just showed you. So how to respond, and actually in the response there are different strategies, a little bit depending on where do you, in the time, where are you located if a shock is occurring. So we can think about more proactive strategies, so that's the ability to anticipate. So then we talk about situation awareness, knowledge, etc. So a lot of work which has been done also in HoliFood, it's really about that forecasting, what will happen. So that's really the proactive strategy. Of course, the question is, to which extent do food businesses have access to all this knowledge and to have access to all these models and predictions? If we talk to them, and this come up later on, surely if we talk about family-owned businesses, I mean, they are not the Nestle's or the Barillas who are here. So how can this knowledge go to them also? So then, if the food safety shock is occurring, then we talk about concurrent strategies, then they have to adapt and they have to respond, and then later on you have more the reactive strategies, the recovering and the knowledge. So what do we learn out of it and how we will do it differently in the next, to be ready for the next shock. So we tried to understand the responses from the food businesses, and we tried to categorize them. As I said, proactive. They really have, they are eager to know, okay, how are this early warning system and foresight tools, are they working? For them, for instance, Rapid Alert is a kind of a forecasting tool. They look every morning what's going on. Yes, so this is more on the proactive perspective. Concurrent, we can think about what do they do if the problems are occurring. This diversified sourcing is very important, so that they are having multiple suppliers. So if something is happening with one supplier, that they can go to another one. And then what was also very important for them, it's about internal collaboration. So alignment over different departments within a business to talk about microbial food safety, because operations, they don't talk in the same way as quality department, as I don't know what department is, so really that everybody knows about food safety. And also, if new products are developed, indeed, that food safety by design approach already. So this is more about collaboration and communication and knowledge sharing within a business. And then as reactive responses, also there, knowledge management and building social capital was very important. So it was about communication, how do we inform everybody post-shock, so when everything was solved, how are we learning out of that? How can we learn from these past incidents? So there you see, it's not so much about technology, it's not so much about high-tech analytical things, it's really again about basic human dimensions, about communication, and about trying not to forget what happened and take this along in future activities. So these were response strategies more at the food business level. Also, if we then discuss with them about the chain level and then more food systems perspective, here that on the one hand continuous and a collective approach indeed to do that forecasting was very important. So that not every business is reinventing the wheel and setting up an AI-driven tool to forecast something, but to have this more together at the sector level, so to avoid that everybody is working again alone. We saw it also with always with food safety incidents. If something is happening, the whole sector or the whole food chain is affected. Think about the ARA bacillus thing some months ago. So that collective approach in forecasting was very important for them. And also again, and then we talk about food safety culture, so to go beyond the technology, but also to adopt more on a human aspects, than about communication, leadership, awareness, that also those dimensions are not forgotten within governing food safety. So to conclude, these drivers of change are leading to unpredicted shocks. Important is to understand which impact do they have. So therefore this DISPER framework can be used. A resilient food safety management system is surely also a flexible food safety management system, but then of course to allow flexibility, a lot of information and a lot of understanding is needed. If you go away from a certain procedure, let's say about time, temperature, which has been followed for years, it's very difficult to go away from that. So therefore a lot of information and understanding is needed. But again, on the chain approach, that collective and continuous approach, as I said, and don't forget about that food safety culture, is so more the human dimensions as communication, leadership, etc. As I said, these tools are available, so we made 2 tools. Our target audience are food business operators, maybe who want to do this exercise for themselves. I can imagine in some management teams it could be of interest to do this, to identify which drivers are relevant for our business, how are they impacting, how could we respond on that. But we are also thinking about sector associations who bring their members together, for instance, the dairy industry, who think on common drivers, who think on common shocks, who think on common potential response strategies, and also risk managers. And I see Emily here from our Belgian Food Safety Authority. We made an exercise in Belgium with the different members of the Food Safety Authority, so the sector associations, to make this exercise for the Belgian food chain. And so the second tool is about resilience strategies. So what could we do to anticipate, to cope, or to respond on those shocks? So they are available on our hub. I think the hub was already presented. Yes, so we also made a guidance document on what to do with it and how to make an interpretation. We also have the presentations on resilience there. So all the information is available in our lifelong learning module on the hub. So this brings me back to thank also the FoodSafeR project. I also want to thank Barilla and Nestlé, because our industry partners, they were also involved during the pilot tests of the tools when we were still looking, okay, how we will do that. So they were a bit our volunteers to try it out. So we got also very valuable input from them. Thank you very much.
All right. Good to see most of you back. So next up we have the poster presentations chaired by Deborah.
Hello. So I have two phones because I rely on our presenters, but I rely more on my alarm. So the poster session will be a pitch of 2 minutes from our presenters. And who can imagine the topic of the posters? No one! Where have you been? We're talking about food safety. Come on. As you know, or should know, food safety is facing a lot of challenges. And we will try to delve into these challenges through the presentations. We will have 12 today, and we start our journey with a well-known pathogen that was mentioned several times these days. Can you imagine which one? Listeria, yes! And Felix Spiegel should present it. Okay. I'm starting the clock.
Now.
Yes.
Okay. Hello, everybody. My name is Felix. I'm a PhD student in FoodSafeR and also at Foxy in Vienna, and I work on Listeria. And Listeria is a major foodborne pathogen and has a high impact on our public health. Furthermore, it has the ability to persist in food processing environments, meaning it sticks around for months and years. And research has identified associations of, for example, mobile genetic elements, gene presence absences, or co-occurring microbiomes that it is associated with persistence. But yet we have no clear idea what's really causing this. And another challenge is that a lot of studies use different definitions of persistence. Meaning different times of isolations, different definitions of what the same isolate is. And in FoodSafeR, we applied a unified sampling and analysis approach to investigate the persistent Listeria monocytogenes. And therefore, we sampled in Greece, Spain, and Austria, and defined persistence as a minimum of three times isolations. That it needs to have to be persistent, and also the same isolate needs to have less than 10 SNP differences across the whole genome. Overall, we sequenced 335 Listeria monocytogenes genomes. We did a pan-genome analysis and identified 364 genes that were enriched in persistent isolates. They were mainly involved in genome maintenance, stress tolerance, and also associated with mobile genetic elements. Virulence genes and environmental resistance genes, however, were more driven by sequence type rather than our operational definition of persistence. And overall, our findings highlight that this is a very complex problem, the persistence of Listeria monocytogenes, and we need to do standardized samplings to build databases. Yeah? Okay. 10 more seconds. Databases to do modeling approaches and identify key drivers and guide future research so that we can make progress on this topic. Thank you very much.
Perfect. Thank you. And we now move to an emerging pathogen from the marine system, yes. And it will be presented by Thijs Koppema.
So, my name is Thijs Koppema. I'm from Wageningen Food Safety Research, and I will show my poster of predicting tetrodotoxin contamination in bivalve mollusks using explainable AI. Tetrodotoxin is a potent marine toxin found since last decade in the Mediterranean, but also the Eastern Scheldt, the North Sea. And it has found that the buildup between locations quite differs. So it has a quite strong temporal aspect, but the temporal aspect of tetrodotoxin has not yet been researched before. So what our idea is to train a model which is able to process this temporal data and then look into the model using explainable AI to look at what could be potential drivers or what is correlating with this toxin appearance. So what we got was data from the Dutch monitoring plan, linked monthly, daily basis meteorological and weather data to it, and trained a long short-term memory model or a temporal deep learning model. Well, this model was performing quite well with an AOC of 0.39, so most of the samples were correctly identified with minimal false positives. And then we used SHAP to look into the model and see at what features it is looking at. From what we found is it is mostly seasonal, so time of day, the sunrise, sun hours, but also more hydrological or meteorological features such as the water temperature or the chlorine concentration of the water. So with this, we can further identify what potential drivers of these toxins are in those fields, maybe predicting when it's not there, and further, making it easier to circumvent it or deal with it. But so the model was able to predict it. The XAI helped us understanding specific predictions, and a couple of drivers were potentially identified. I'm done.
Thank you. Okay, so predictions requires data, and Floor van der Maar will present one of the largest database available in Europe.
Thank you so much. So my name is Floor van der Maar, also Wageningen Food Safety. And in the EU, every member state reports their monitoring data to EFSA. And EFSA shares this food safety monitoring data again on Zenodo, which is really nice, free data, nice to use. But they share it in a file that there's one CSV file per contaminant type, per country, per year. So that ends up being over 1,000 different little type files. And with over 400 gigabytes worth of data. So not actually that easy to work with. So what we've done with our data science team is that we've created one big harmonized data set out of this by merging all of these files and harmonizing them and coupling them to ontologies. So there we ended up with the CHEFS database, and CHEFS stands here for Comprehensive European Food Safety Database. We published a paper about this, which you can access via this QR code, which I hope works via poster and screen. Also what we're doing right now is that we're developing a dashboard. Well, first thing, you're free to download the CHEFS database yourself. You can access it via our GitHub, but still, it's pretty big if you want to use it in its entirety. So we're building a dashboard about it that you can make all of these nice little visualizations yourself. And also that gives you the opportunity to download subsets of the data. For example, if you want to know something about mycotoxin in grain in a certain year, a certain country, you can export your own CSV file about this. But yeah, or pesticides in oranges, whatever you wish. It's free for you to do. The nice thing is we've seen a couple of examples of that being developed in HoliFood as well. Having a database big like this with over 400 million entries makes this really very useful to use for AI machine learning models since it's really big. And that was it. If there's any questions, please find me during the drinks.
Thank you. So once a risk is detected, the next challenge is tracing its origin. And Alexandra Irgang will present two posters on behalf of the team from the German Federal Institute for Risk Assessment.
Yeah, so. Thanks, speakers. It's not my poster, and I'm here on behalf of my colleague. I have to stick to my notes here. I'm sorry for that. So Food Chain Lab is Food Chain Lab Web is a free open access software which supports the food safety authorities in tracing food and feed through complex global supply chains during foodborne disease outbreaks or other contamination events. over the past years, FoodChain Lab Web integrated contributions from various projects like FoodSafeR corporations, the One Half EGP project, Cohesive, or the FoodSafeR project, and creating a modular integrative platform used by food safety authorities such as the Austrian AGIS or the US FDA. The FoodChain Lab Web offers interactive data visualizations, analysis, and reporting. The supply chains are visualized as networks with nodes and edges and can be interactively rearranged. The key information on food business operators and food items is displayed in data tables with advanced filter functions for in-depth analysis. Hypothetical cross-contaminations or geographical clustering events during outbreaks can be simulated using a scoring algorithm for deliveries and operators. And it's possible to automatically optimize data on food business operators. As a most recent feature, FoodChain Lab Web now includes functionalities for collecting tracing data via standardized Excel sheets. Yeah, so that's all I would say. It's a comprehensive, it's easy accessible, everybody can use it. It's free available, and it's connect tracing tools on local, national, and European levels. Yeah. Person here now. Oh, I'm so sorry. Nobody said this. I'm sorry. So now this is the poster with the FoodChain Lab, and the other nobody can tell me. The other one was, I'm sorry, on the interactive, on a review, this one. Thanks a lot. Sorry for that. So this post is on a review conducted within the FoodSafeR project to provide an overview over determinants on local and global food chain supply chain, global and local food supply chain, sorry. And it was conducted using a driver's approach to examine the driver's contribution to emergence of food safety risks, and included a qualitative review of scientific and trade literature focusing on 8 determinants that are the food safety, technology, environmental protection, economic growth, sustainability, traceability, and data management. And the goal was to identify distinctions between global and local food supply chains. So as a result, it revealed that the products in the local food chains were perceived more sustainable and environmentally friendlier than products in the global food supply chains. But in contrast, global food supply chains were considered to provide greater food security, enhanced safety measures, adoption of advanced technology, and significant contributions to economic growth. It had higher levels of traceability and integration of more sophisticated data management systems. So in conclusion, further research is needed to provide evidence-based support in optimizing the supply chain, the local supply chain, via harmonized data structures or software systems that are needed. So I'm so sorry, but yeah, you may have a look on the FoodChain Lab poster afterwards.
Okay, this is the beauty of the live. Now, once Ksenia, yes.
I saw my poster, so.
Yeah, indeed. Please welcome Ksenia Paskari.
Hi, my name is Ksenia Paskari. I'm a researcher at the German Federal Institute for Risk Assessment and in Berlin. And as part of the HoliFood project, our goal was to use AI on high-resolution mass spectrometry data to perform untargeted identification of new and existing chemical hazards in maize. Imagine receiving thousands of signals from high-resolution mass spectrometry experiments. And before artificial intelligence can learn from that, just like Marco mentioned in his presentation, we need to extract the data in a compatible format and ask a very critical question. Are the extracted signals real? Are they reliable? And are they biologically meaningful? Today, I bring you a 6-step workflow that brings us from the raw data files towards a clean feature table that is ready for downstream data processing. After acquiring our data, we created also, to streamline all this, user-friendly interface, our Hermes processing platform, that performs feature extractions, combines the feature identified in different batches, and after subtracting the feature identified in blanks, it performs and applies a comprehensive peak cleaning algorithm that identifies and removes low-quality peaks in our data. And because we worked on different batches, we also performed a batch correction within the batch based on the feature occurrence in the QC samples, and we performed an intensity drift correction between the batches based on the reference samples that were injected with each batch. In the end, we performed also a comprehensive data inspection of our feature table in order to assess the structures of the data, identify outliers, and verify if all the cleaning and filtering processes that we did until now provided a coherent feature table. In the end, it is not only a processed data, it is a curated feature table that presents a solid basis for the identification of chemical hazards. So if you want to know a little bit more how we studied our data, please find me in the next breaks. Thanks.
Thank you. Okay, so once data are generated, AI can help us to uncover patterns, things that otherwise will stay hidden. Andrea Carnoli.
Thanks. Okay, so my name is Andrea. I work at FoodSafeR in the data science team. And you probably already see this poster, so, well, through a presentation from Marco. So we start this pitch by asking you a question. We talk a lot about foundation models in the first day, big databases, but what happens when we don't have a lot of data for AI? Well, it's not that the AI will explode or your computer will be tossed away, but just the performance of the model will be unreliable. And that's what we observe with in this project with the first confusion metrics that show the results that look nice, but actually they're not very nice because the model is very influenced by batches effect, like day of analysis. And by doing transfer learning, which basically consists in making the model a bit aware of this type of data with a bigger data set, you can improve your chance of having a useful model. And the curious thing about this work is that it doesn't matter if the initial data is a bit far off from your fine-tuning data, as we discovered in this work and in a bit of more case studies. So we applied this concept to untargeted LCH-RMS data, and we found out that by pre-training our model on cow urine and where they measure steroids, we can fine-tune it with chicken muscles, where we analyze unknown chemicals. Well, we know some because we spiked the samples to be able to judge the model, but they were two very different data sets. So yeah, transfer learning is very nice. AI always give results, which can be misleading if the data are not enough. And yeah, if you have a bit of any questions, you can find me around for the drinks or something.
Thanks, Andrea. Okay, now Selcan Semerjots will explain us the barriers in AI implementations.
Okay. Thank you very much. Hello, everyone. Good afternoon. This is Selcan from Wageningen University Food Quality and Design Group, and I will briefly present the results of a master thesis that explored barriers to the implementation of AI in food safety monitoring. And AI is a promising data-driven tool to enhance food safety monitoring, as we all know in this room. And however, its implementation in industry for food safety systems remains limited. Therefore, we wanted to better understand what prevents its wider use in practice. So we conducted a semi-systematic literature review and in-depth expert interviews, and we analyzed the data thematically by using techno-managerial approach. So we produced categories for the barriers. And both in the reviewed studies and expert interviews, mainly technological and technical barriers, especially data-related issues such as data availability, quality, and the volume of data emerged. Besides, the experts mentioned the limited interpretability and explainability as an important technical barriers for AI models. I think this one also mentioned in one of the presentations yesterday. Unlike human experts with AI models, sometimes it is difficult to understand how decisions are made. So this is also emerged through the interviews. Interestingly, managerial and organizational barriers emerged only through the interviews. This may be because many reviewed studies are still conducted at design or lab scale. So therefore, managerial or organizational aspects are overlooked in the reviewed literature. Within this barrier category, human-related barriers are the most mentioned ones, such as limited knowledge, awareness, and skills among the employees, etc. Of course, the economic and resource constraints, particularly for small and medium enterprises, as well as coordination and collaboration-related barriers emerged. Also, the lack of standardization for AI integration was mentioned as a key barrier in both literature and the expert interviews. And experts further addressed a lack of harmonization across the countries and regulatory lag, which means the technological improvements are generally one step beyond the regulations. And data ownership issues, of course, one of the barriers they mentioned. Overall, the results of this study contribute a more holistic understanding of AI implementation barriers in the food industry and show that addressing these barriers requires multidisciplinary approach. Thank you.
Thanks. So, technology is not the only way to address the problem. Citizens can also contribute to early detection system. So welcome Tamara Spivey from Newcastle University.
Hi, I'm Tamara. So this work kind of stemmed from Work Package 5, where we were looking at citizen science and how we can bring that into early warning systems. And we were looking at centralized versus decentralized citizen science infrastructures. So we were comparing, essentially, we needed a case study for this, so we were looking at the yellow-legged hornet and the different monitoring systems in different countries within Europe. So we looked at the UK, Italy, France, and Germany, as these all have quite different monitoring systems, we discovered. So we had kind of, myself and other colleagues within HoliFood, we looked at, analysed these different reporting systems and the platforms and all the different documentation that came along with it, and then compared how these infrastructures were different. So we looked at the reporting structure, the verification pathways, and the response coordination. So this ranged from national infrastructure platforms to quite regional and state-based platforms. So UK and Italy, we found, were quite nationalised and very centralised. And then it went down to France was a bit more fragmented, and then Germany was quite regional and even more fragmented than France was. So essentially, we then looked at this and then kind of looked at the verification processes and how easy that made getting these citizen science reports that were given through applications into actual reporting systems so they could make a difference. And we looked at this and the verification process, and my brain's gone. And basically what was most important. So we found that the centralized infrastructures were the better ones for getting things more into the system, and then verification was key. in ensuring that everything was correct, because it's still citizen reports, so you have to kind of filter through that noise. And then response coordination and integration was really important as well. So then we wanted to link this back to emerging food risk, and then how we can kind of deploy this throughout Europe and the rest of the world to bring direct citizen science reporting kind of more into these early risk warning systems.
Thanks. Okay, understanding risks is important, and it is important to know how people perceive them. So, Novieta Sari from Newcastle University.
Hi, thank you very much. To be honest, I never talk in the 2 minutes, so I don't know how to manage this, but I give a try. So as you say, that's this poster basically is a study that we are conducting at 4 different European countries, UK, France, Germany, and Hungary, interviewing parents with children aged 0 to 5, because as we might know, climate change has reshaping the children's food safety on the children's plates. So, on our study, we're collecting 80 respondents, 20 participants from each country, and then we're using a semi-structure, and because we found that parents, even though they are aware there is a climate-related food safety, but yet they still quite passive to do some actions. And why? To explain that, we're using construal level theory, which show that psychological distance is happening across 4 dimensions, including temporal, spatial, social, and hypothetical. So probably from your the image over there, you can see what's the psychological distance that's occur amongst parents. So most of them feel like that's the climate change is a future problems. So the mindset of not me, not now, is being creating and shaping in the mind of the parents that's influence their decisions making in how they are seeking the information, sharing, and also perceive and identifications the issue of the climate change. So that's also happening because the climate change apparently is being framing not as a public health concern, but is more on the sustainability issue. And then also some of the fragmented policy occur also that's creating people becoming more appetite to the issue of the climate change. So yeah, that's it. Thank you.
You did it. Okay, perceptions are not always the same across group, and that's why inclusion and diversity are important. And welcome Margaret Vandenberg from Wageningen University.
Yeah, thank you so much. I guess I'm the last one, yeah? Yes, well, get the last one. No, okay. Okay, well, you see the poster on the left side. It's the part on the collaboration internally in the project. And the right side is more what we did within VP's work packages. So to work with the living labs to make the guidelines inclusive, and for the results of the citizen part, especially the survey. And of course, compliments to you in the communication, especially on the International Day of Women and Girls in Science. There were always posts, and we worked together there. I would love to make some remarks on the recommendations, because you probably will make new projects again. I think it's very important. It's an EU requirement that we include the gender dimension. And it's also a criteria of excellence. So you make points there or you lose points there. And it's good to see that gender component and the people working on not as a police, we don't want to do that, but as a co-creator, colleague. And what I found out in not only this project, but also other ones, is that most consortia in Wageningen are well balanced. the survey, what was a kind of audit, what are the capabilities and the ideas and the perceptions of the people into the consortium, well, it turned out there's a lot of awareness, but there's much to learn and to improve. You can see all the results on the website under gender dimension, and I have some copies here of the posters if you like to take it. And then what I think it's very important is that if you make new projects, it's really better to include somebody with knowledge on gender from the beginning in the design, but especially also the budgeting, not only for that person. I had only 1 month. It's not the management's fault. There's the third group of managers who actually did the project in the end. But we all need time to exchange, to find out what does it mean, how can we do it, and also have some workshops, which also gives a lot of good things to the team building and the internal communication. And that means for the cooperation, but also the integration in the work, the research, the concepts, the operationalization, the method analysis, and of course the interpretation and the communication, even the imaginaries. Okay.
And that's it. We finished the symposium. And I want to thank all the poster presenter. And thanks Nathan, Rudy and Martin, Naomi. I can be emotional, so Nathan, this is the microphone.
Yeah, thanks everyone for being here. Just from my side, a few household messages, and then Rudy will give a short summary of what we have learned in the past 2 days. I also want to thank, first of all, the people here and the speakers, as well as the people online, because there has been a live stream. There is also a recording available afterwards, so if you want to see all the presentations again and again and again, that is possible. I want to repeat the message about leaving the name tags if you're not going to collect them at the entrance so that we can dispose of them correctly. For those, especially from the projects, if you haven't written or signed in the sheet yet, please do so, because otherwise your travel costs will be flagged and not reimbursed, so you don't want that. Anything else? No. Thank you all. So there's a few more months remaining for both projects. Looking forward to those. We got a couple deliverables, just a few to check. So we're going to be okay. Thank you all. And from my side and on behalf of HoliFood, have a nice day and a good trip home.
Yeah, thanks, Nathan. So it's again an old guy, an old white man who finalizes things. Well, dear friends and colleagues, I have indeed now the great privilege to officially close this, I think you agree with me, inspiring conference, but certainly not before having said a few words to you. I hope that you all do agree with me that organizing and also joining this joint HoliFood FoodSafeR conference has indeed been a time investment, maybe also for some of you also financial investment well worthwhile making. I believe that the motto of our event, Safe Food Smart Future, has been impressively demonstrated with a great number actually of novel AI-based tools, for instance, for the improved prediction of emerging hazards and associated risks, both pre-harvest and also post-harvest at the retail level, but with a number of novel diagnostic tools and also for on-site detection for natural toxins and other chemicals in our food. Some impressive case studies, I think, have also been presented these 2 days. On top of that, we have learned, I think, a lot about the newly identified drivers and also stresses of our food systems, particularly today. Another, at least from my point of view, important piece of information related also and relevant to the food and feed industry And please also allow me to also conclude that the overlap, the observable overlap between the two projects, HoliFood and FoodSafeR, can at the worst be considered as healthy overlaps. I believe that we have indeed been pursuing complementary approaches, and that we have also been leveraging synergies actually to the extent possible. Things we are now living in very complicated times. There are consortia agreements and there are grant agreements, and there is all the IPR issues around that. So certainly there are some limitations, but also particularly this conference, I think today has indeed demonstrated there's a good spirit between these conferences, between these two consortia, and also with other consortia such as Food Safety for EU in the past, Food on Food in the past. So we have really, I think, made an effort to leverage also knowledge which has been generated before. And I think that is really good news for the taxpayers, because in the end we have to justify, and that is also why I'm telling particularly younger people, don't be shy. It is really also our duty to make our results visible, and that I'm referring both to SCI publications, but also visible vis-a-vis the public, like LinkedIn platforms whatsoever. You can also go on Instagram and make some nice videos whatsoever. But I think that's also from a taxpayer's point of view something which we have to do, and it's I think also in the interest of the consumers and also of the food and feed industry. There is also great potential for post-project exploitation, I think, of our results. There are still a few months to go, but there is really a lot of I think, potential there. And this certainly includes the before-mentioned novel AI-assisted holistic approaches for food system risks, also and for predicting these risks, as well as, for instance, for the FoodSafeR digital hub, which has been designed particularly for the stakeholders along the food chain from around the world, also including our advisory board, actually covering all the different hemispheres, so that we can also consider what's going on in the world and also have this constant feedback loop. And we will certainly make an effort to maintain this system. We have actually been successful in previous projects by actually asking companies who have licensed our platform, and we will definitely go into bilateral dialogues with the industry and also with stakeholder organizations. So at least within this room, so sign up and let's stay connected also via these platforms. There's HoliFood platform, the FoodSafeR platform, and several others as well. Finally, I would like to express our great gratitude to Nathan, so there are different pronunciations, and you have really a dream team here, and it was really a great pleasure to collaborate with you, and thanks for facilitating this, particularly in this fantastic room at the premises here of Wageningen. I would also like to express my thanks to all the members of the FoodSafeR as well as the HoliFood team, and it has really been a pleasure to collaborate with you. I'm also glad that Ine was able to join. I wish her, I don't know whether she's still in the room, to join, and I wish her really a full and quick recovery. Was really glad to meet her, so please forward my regards to her again. It looks like an obvious and very natural thing that we have such a fantastic cooperation, but this is not something you can take for granted. Remember, we have been competing consortia, and I can tell you from my own life, I have a long history also with European projects, there is a tendency that you make more enemies than friends, because there's always somebody who is out and somebody who have to kick out maybe on the way to a project. So it's really a tough thing, and the competition is getting tougher and tougher, and particularly against the backdrop of this competition, it's great to have this friendship and this great collaboration. Special thanks certainly also to all the lecturers and also to all the colleagues who have very ably presented the posters here. This was also very enjoyable. So there's the next generation already taking care of the emerging food safety hazards and associated risks. So thanks also for that. And last but not least, thank you all for coming. That being said, thanks for joining. Keep in touch. Have a safe trip home, and see you around, see you in the future, and particularly for the FoodSafeR consortium, and also in the name of Martin Wagner. See you tomorrow, because we have the annual meeting tomorrow. Maybe for the HoliFood you have a similar thing. No, well, you're done. So see some of you tomorrow. Thank you very much, and take care.